Okay, next. Dr. Bansari.
Hello everyone, myself Dr. Bansri. I am from MP Shah Medical College Jamnagar from Gujarat.
Good afternoon, everyone. Myself, Dr. Indira Gore. I am a third year pathology resident from Grand Government Medical College, Mumbai.
Good afternoon. Myself, Dr. Nekibali Hazarika, third year PGT, Guwahati Medical College, Guwahati.
Good afternoon, everyone. Myself, Dr. Nisha Chauhan, third year PG at Government Medical College, Bhavnagar.
Good afternoon, everyone. I'm Dr. Rahul Skotra, third year PG from Armed Forces Medical College, Pune.
Good afternoon, everyone. I'm Dr. Soumodip Das, first year pathology resident, Bardhaman Medical College, West Bengal.
Good afternoon, everyone. I'm Dr. Anvesha Datta Chaudhary, first year resident, All India Institute of Medical Sciences, Bibinagar, Hyderabad.
So these are our participants. Next is the reminders. The important one here is that please do not send requests for annotation to the presenters. These are the disclaimers. Few more. Our next lecture will be on cervix and human papilloma virus by Dr. Radhika, who is ex-professor and head in cytology and gynecological department, PGI Chandigarh. This lecture will be on 10th January, this is Tuesday, followed by a slide seminar on 12th January on gynecology cytology cases by Dr. Kamal Meher Bano, who is professor in pathology, GMC Nagpur. So our upcoming workshop will be on 4th of March of this year on molecular techniques including NGS, which will be done by our team, which is headed by Dr. Nikhil Patkar, the Tata Memorial Center, Mumbai. So without any further ado, we'll start our lecture.
So should I start?
You, sir, you can share your slide.
Yeah, I'm sharing. So hi everybody, good afternoon. So as you, today me and my team and myself would be presenting you the basics of cytogenetic techniques. And I would like to thank Dr. Bojes and Dr. Gujral for providing this platform for us to share our experience with all of you. And so without much delay, I'll just start. So we have a series of talks here. We have about 4 to 5 talks. So the first talk would be by me, where I'm just going to introduce certain basics of genomics. Then we will have Samipa, who's going to talk about fish technique as to, how do we go about fishing in genomics, which will be followed by the different types of fish probes by Arun. And then we have our very bright fellows, Dr. Aishwarya and Karthik, show you some interesting cases just to, impact on, put an impact to see how on a day-to-day diagnostic lab, cytogenetics is used to bring about a change in the management of cancer patients, right? So I'll ask the most common question asked in the COVID era. Can you see my screen and can you all hear me properly?
Yes.
So this is the disclaimer for the presentation. And to start with certain basics, So if you look at DNA, right, and so we've been working, so generally the cytogenetic department deals mostly only with the DNA, we do not process any RNA, right? So if you look at the DNA, it just has only 2 jobs. Number one, it serves as a store of information. And it is its job to ensure that this information is passed on to each new cell upon division. And second is it directs the synthesis of proteins by transcribing to RNA and then translating into proteins. Yeah. What do I do this? Should I prove? Huh?
No, don't approve, sir, please. Oh, that is an annotation request. Don't approve.
Okay.
Audience, please.
So to all the participants, please do not send annotation requests like Dr. Priyanka requested. Do not do that because then it creates a lot of problems for the presenter, please. Thank you.
And the second role it plays, I said, is it synthesizes protein. Now, if you look at the structure of DNA, it's a very dynamic structure. The DNA, based on its function, can keep changing its structure. So in an interphase nuclei, the DNA is in the form of a thread, right? Because it is storing all the information, both genetic and epigenetic, that makes a person or a cell what it is. And as I said, one of the rule is for this DNA to transfer all the information to the daughter cells. Now, it is not possible for this thread-like structure to be transferred to the daughter cells. So what happens is when the cells start dividing, the DNA starts condensing itself. And in the process of condensing, it forms what are called as chromosomes, right? So what is a chromosome? Chromosome is nothing but a molecule of DNA, right? It takes up a chrome, we stain it, and that is why it is called a chromosome. So as I said, the DNA has different layers of arrangement. So it can be in the form of a thread, it's a double helix, it moves around the histones, and then as it condenses further, it forms a chromosome. A chromosome has a short arm, which is called the P arm. P is petit in French. And then it has a Q arm, which is a long arm. Why do we call it Q? Because Q comes after P, that's it. So just for that, right? Now, before I go, I thought I should share with all students this interesting story about the structure of DNA. Now, this was an era when all scientists in the world knew that whoever discovers the structure of DNA will get the next Nobel, right? So this is basically a story of these four people you see in the picture: Rosalind Franklin, Maurice Wilson, and of course Watson and Crick, who actually finally have been credited for discovering the structure of DNA. Now, okay, let me put this as a pointer. Okay, so Maurice Wilkins and Rosalind Franklin worked together, and Maurice Wilkins, this guy here, you see, he was the senior. And when Rosalind Franklin joined, it was hate at first sight. They just hated each other. So now you have two colleagues, and both of them had been given the job of doing research on the DNA structure by the head of the department, and they just hated each other. They just couldn't get well along. At that point of time, what they were doing was they were doing what is called as an X-ray crystallography to look at the DNA structure. So it is nothing but, whenever you put a light on an object, it forms a shadow. Now you can analyze the shadow, and based on that, you can say what the object is. So what they were doing was they were taking DNA and they were bombarding this with those X-rays, and they would then analyze the shadow to see what the DNA structure is actually like. But as I said you before, the DNA structure is so dynamic. So while Rosalind Franklin was very good in technique, she was able to, do the wet lab very well. Maurice Wilkins was excellent in data analysis. But yes, they never spoke to one another at all. So at the same time, Watson and Crick got along well. They got along well right from the day one. They would often go drinking to the pub near the institute. And so one of the days when Rosalind Franklin was on leave, Maurice Wilkins opened the drawer and just showed them this picture which Rosalind Franklin had taken to Watson and Crick. Meanwhile, Watson and Crick, they were thinking their theory was that DNA is a triple helical structure. But the moment they saw this image, they knew that the DNA was double helical structure, right? So Maurice Wilkins quietly, without Rosalind Franklin being there, showed this picture to Watson and Crick, and then they knew, Watson and Crick knew, based on that image, they actually made the double helical structure. And the first thing they did was they went to the Eagle pub, and there they, announced that we have discovered the secret of life, right? So if you visit this pub, you will still find this plaque where it's mentioned that the first announcement of the discovery of secret of life or the DNA was made in the pub by Watson and Crick, right? Now, three papers came that year, and Maurice Wilkins, Watson, and Crick were given Nobel Prize. Meanwhile, Rosalind Franklin died of ovarian cancer because of the exposure she had to the X-ray while she was taking the pictures of the DNA. So she never got the credit. And much later in 1999, Watson said that I didn't steal the image; I was shown. So I always feel, that the department, if you fight in a pub, it is better than fighting on the PubMed, right? So you'll have better success to produce much result if you focus on the pubs rather than the PubMeds. So I just thought I'll share this story to say how important it is to work together. Now, what happened was what the image you see here is a network of papers published in the science journal in 150 years. Now, each of this dot is a paper. Okay. Now, each of this dot is linked to another dot, which is again a paper that has been referred. Now, this is where the Watson and Crick paper came. Okay. And anything below this was the papers that were referred by Watson Crick and the reference of those papers. So before this, there were not many papers in genomics at all. But the moment the structure of DNA was discovered, here you see the volcano of papers that came in, clearly showing that this was one of the most important landmark in the study of genomics, and that completely changed how we understand the genome today and how we analyze it. So when you look at the genomic techniques that are available in a lab, right, you have karyotyping. We will talk about it in detail as we move ahead. So karyotyping is looking at the chromosomes. FISH is fluorescent in situ hybridization. Single nucleotide polymorphisms are nothing but a digital karyotype. And of course, there are a whole lot of molecular techniques which you will be dealing with in your next workshop. So karyotyping, actually, you're looking at the whole genome of a single cell. You can look at the whole genome, whereas FISH is more targeted. Anyways, we'll talk about it. So being a pathologist, I like to look at these techniques as those that are microscopic based. So karyotyping and FISH, you use the microscope. You're looking at the genome with your own eyes. So, FISH, you can actually look at a gene with your own eyes. So it's pretty exciting. It's a beautiful world below the microscope there. But there are abnormalities in the genome that cannot be identified by the human eye on the microscope, and that is where you have submicroscopic abnormalities. So when you look at the submicroscopic abnormalities, you have the copy number alterations and you have the mutations. Now, one of you may ask me, what do you mean by copy number alterations? Because when you read your books, you will see the short forms as CNA or CNV, copy number variations. So, God has blessed all of us with a diploid genome, that is 46XX or 46XY. Any loss or gain of genome genetic material is a copy number abnormality. Gains of number of chromosomes is a copy number abnormality. Loss of a chromosome is a copy number abnormality. Deletion of a part of a chromosome is a copy number abnormality. Deletion of a gene is a copy number abnormality. Deletion within a gene is a copy number abnormality. Duplications are copy number abnormalities. And mutations, you use molecular techniques. You can either go targeted or you could go in a genome level. Now, there is always this confusion, which lab does what? So cytogenetic labs do karyotyping, FISH, and arrays. And the molecular diagnostic lab goes much deeper into they look at the DNA, the RNA, the PCR, and they look at sequence mutations. So molecular cytogenetics is nothing but we have actually techniques like FISH and SNP arrays are molecular cytogenetics, wherein we have combined the knowledge, we've married the knowledge of molecular, that is the nucleotides, to the cytogenetics or the chromosomal abnormalities, and then you can use it. The advantage of molecular cytogenetics is you can apply it to fixed archive tumor material. It is accurate, it is specific, you do not need live cells. So if one of you want to do some FISH on a breast tissue, say 5 years back, you can take your blocks and you can do molecular cytogenetics to study it, right? So these are the molecular cytogenetic techniques. FISH, MFISH is multicolor FISH, spectral karyotyping, comparative genomic hybridization, and SNP arrays. So what is the difference between karyotyping and FISH? Karyotyping, you can screen all the chromosomes. It is one of the simplest technique to look at the whole genome of a single cell, right? Whereas FISH is more specific. It answers the question you are asking based on the probe you use. So if you ask, well, is BCR-ABL present? You use a BCR-ABL FISH probe and say, yes, it is present or it is not present, but it doesn't tell you what is happening in other parts of the genome. But karyotyping will tell you what is happening in other parts of the genome. You can pick up a 922 and you can pick up additional abnormalities as well. That is where the karyotyping has an advantage. But FISH is more rapid, it is quick, and will answer your specific question. Now, conventional cytogenetics is nothing but karyotyping. So as I said, the DNA can be seen as chromosomes only when the cell is dividing. That is why you can see the chromosomes only during the metaphase. So that means you need metaphases to study chromosomes. Now, to get metaphases, what do you need? You need live cells. You need cells that have the capacity to divide. If you have dead cells, you cannot do karyotyping. That is why the molecular cytogenetics has an advantage there, wherein you can actually fish or you could extract the DNA from the dead tissue in the formalin fixed blocks and you can do your studies. Whereas for karyotyping, you need live cells. Now, you not only need live cells, you need cells that can divide because only when a cell will divide, it will produce a metaphase. So in order to get that metaphase, you need to have cells which are proliferating. Now, there are certain cells in the body which are spontaneously proliferating. For example, the bone marrow, the lymph node. So we have done FNACs on lymph nodes of patients and put them to karyotyping and identified abnormalities. Solid tumors, you can do karyotyping, but it's very cumbersome. You need fresh tissue. So somebody has to go to the OT, and before the surgeon dips it into the formalin, you have to collect the solid tumor. There are huge issues of contamination. That's a big problem. And of course, in prenatal cytogenetics, they do chorionic villus biopsies, and even that has a proliferating cells. Now, certain cells in the body, if you want to study, they are already matured and will not spontaneously proliferate. For example, the peripheral blood lymphocytes and the plasma cells in multiple myeloma. So, what do? You stimulate these cells to divide. So, you use PHA or phytoheme agglutinin to stimulate lymphocytes to start undergoing division. And plasma cell, you can use interleukin. So, you have a patient of myeloma, you want to do karyotyping. How do you do it? Plasma cell will not divide spontaneously. So what you do is you take interleukin cocktail, you put it in your sample with plasma cells. Your plasma cells will be stimulated to divide. And once they are dividing, you can arrest them in their metaphase. Okay. So this is a question for all of you. What is the preferred anticoagulant sample to be sent for cytogenetics? Is it sodium
Heparin.
heparin?
Sodium
Sodium heparin.
Okay, so it is the sodium heparin. Why not EDTA? See, because EDTA chelates calcium, right? So calcium is essential for all processes in our body. So your cells will not divide. Okay, you may say, okay, why don't we do fish on an EDTA sample? Yes, can be done if there is no choice. But EDTA are crystals. So these crystals will produce a lot of background fluorescence and can make the interpretation of your fish result a little more difficult. Sodium heparin is best, and lithium heparin, again, lithium is toxic to cells, right? So basically, peripheral blood and bone marrow, you have to use preservative-free sodium heparin. Please don't freeze the cells. You will kill them, right? They have to be in about, so you'll say, okay, how do I transport these samples? So they have to be at around 4 to 8 degrees. See, generally, when you read the Western books, they say room temperature. That is the room temperature of Europe, right? 15 degrees, 18 degrees. Room temperature of India in summers is 45 degrees, right? So then your sample will not work. So you can always put them in an ice pack so that the temperature is around 4 to 8 degrees and it can be sent for karyotyping. The first few ml of the bone marrow contain the highest proportion of cells. So at a diagnostic time point, it's good to have the first pull coming to cytogenetics. Now, what are the solid tissue samples you can receive? You can receive skin biopsies. Now, when do we do skin biopsies? When you want to do karyotyping of skin fibroblasts. So the skin fibroblasts can be made to divide. The skin fibroblasts are done usually when you have to rule out mosaics. You can send chorionic villi, products of conception, and of course tumor tissue. There's a big issue about microbial contamination in these tissues, and they have to be transported in a sterile vessel containing growth or culture media and not formalin. So I'll show you how do we go about the bone marrow. So at the end of my presentation, what I'm going to do is I'm just going to show you quickly a video how it is done. Okay. So you take the bone marrow or the blood, then we start culturing the cells. So in order to culture, we put them into an RPMI media, right? So we have the cell culture media, which has all the nutrients the cell needs to divide, okay? And then we put it in a 37 degrees with 5% carbon dioxide. Why carbon dioxide? Because carbon dioxide, when the cells are, they are alive cells, they are dividing, they would be metabolizing, so they would be a release of carbonic acids. So in order to maintain the pH and maintain the optimal quality for cells to divide, we use a carbon dioxide incubator. Now, when these cells are dividing and when they undergo metaphase, we add colcimate. What is colcimate? Colcimate will arrest the cells in the metaphase. It will poison the spindle fibers and then the chromosomes stay in the metaphase. We add a little hypotonic solution so that the cell swells up, the chromosome gets separated, we fix it, we make a slide out of it, we stain it, and then you have stained chromosomes on a slide. And then what we do is we capture images of these chromosomes using a microscope and then we analyze them. So this is what we do. You culture, you harvest, you make slides, you band, and you analyze. Okay, which of the following samples would require stimulation to make the cells divide for karyotype analysis? Amniotic fluid, chorionic villus biopsy, bone marrow, or peripheral blood?
Peripheral blood.
Because the peripheral blood will have lymphocytes, they are mature cells, they have to be stimulated to be dividing.
Yes, sir.
Now, please know that peripheral blood, the culture is about 69 to 72 hours. So you should know that, because if you are sending a sample for peripheral blood karyotyping, so basically we use phytohemagglutinin, which stimulates the T cells. So the metaphases you get in peripheral blood are coming from the T cells of the blood. So it will at least take 3 days to get metaphases, right? So you must know that. Bone marrow, we just harvest about 16 to 18 hours. Amniotic fluid is 2 weeks or more, and it's a very cumbersome process to do that. Now, why bone marrow in 17 to 18 hours? Because if you look at the cell cycles, right, you have the interphase and you have the mitosis, yeah? Then you have this S phase, okay? So this is where we need the cells to be in metaphase when we are looking. So for a cell to undergo a cell cycle, it takes around 17 to 18 hours. And that is why in a bone marrow, what we do is we culture them, we add colcimate. So every time a cell divides, it is arrested in its metaphase because of the action of colcimate. And then you will get a large number of metaphases. So during which phase of mitosis is DNA replicated?
Interface.
Yes, the interface. So as I said, the cell cycle is about 18 hours, and that is why the bone marrows are put for 18 hours culture, and after 18 hours, you will be getting your chromosomes. Okay? We basically use 2 different cultures. It is important to use 2 different tubes, because if one gets contaminated, it can be an issue. So in one, we add colcimate right in the beginning. In another, we add colcimate the next day morning, so that every time a cell divides, it's arrested in its metaphase, and then we get its metaphases. Which stage of cell division are chromosomes best studied?
Metaphase.
Now, please understand, there are very strict protocols that have to be followed, because a small change in protocol, I'll give an example. This is how we want our chromosomes to look, right? Each chromosome is individually seen here. If you do not follow your protocol properly, this is how your metaphase can collapse. Now, it is impossible to study the chromosomes here, right? Now, when we get the chromosomes, we look at the banding. Now, what is a banding? It's a part of a chromosome that can be distinguished from adjacent segments by appearing dark or light. So the GIMSA stains actually stains this chromosome. Please also understand that the light bands are transcriptionally active. They are the more active zones, and the dark bands are not transcriptionally active. So it is not just the look, but it is also gives an idea about the functional aspect. Now, when you look at the pattern of the bands, it is just like a barcode you see in when you go for shopping. So you scan a barcode and you'll get a product. Similarly, you scan a chromosome, you will be able to get your RNA and the protein. And each of this chromosome has a unique pattern of light and dark bands. So you can see this is the microscope here. We have a camera here. We have a slide. We are capturing the pictures of metaphases. And this is the computer with the software attached to it. Now, what you see here are interface cells. What you see here are metaphases. Can you see this? So these all are metaphases. Now, once we have the metaphases, as I said, each chromosome has a unique pattern of light and dark bands. We use a software, we identify each chromosome by its banding. So it's just like morphology. And then we place them first, okay? Now, this is a karyotype of a patient of acute lymphoblastic leukemia with 77 chromosome. Now, based on the banding pattern and the position of centromere, the chromosomes are first identified. They are placed. And then we look at the banding pattern to see if there is a change in the banding pattern. So are there deletions, translocations, and so on, So now, so far, each one of us have our own favorite. Like I like chromosome 7. To me, it looks like a guy wearing a cap and a bow. One of my students loved chromosome 9. According to him, he said, looks like a petite lady wearing a a two-piece swimsuit. So, well, anybody can use their imaginations, but what you do is you just arrange the chromosomes, you look at the banding pattern, and you identify the different abnormalities. Okay. So, somatic cells have how many chromosomes? And
46.
46 and
46
diploid.
Okay. So as I said, we look for translocations. You can see we have a translocation of chromosome 8 and 16. Ashwarya later will tell you an interesting story about this patient. Sometimes the abnormalities are very subtle, So like the 3q abnormalities are very subtle. Now, this is a 1119 translocation actually. What you see here is one dark band here, that's all. So you need to really, your life is kind of a little, is all about shades of black, white, and gray here. And then you look at these subtle changes and you identify the abnormalities. So this is a translocation 1119 MLL rearrangement, which is so important to identify because MLL is associated with high risk in leukemias. Okay, now, karyotyping can, about 5 megabase pair abnormalities it can identify. Now, there are very small deletions which cannot be identified by karyotyping, which cannot be identified with human eyes. So for that, you need to use other techniques like FISH. So which of the cytogenetic technique will identify a micro deletion, a small deletion?
Inversion analysis.
So actually you can use FISH, right? Because FISH you can go for small micro deletions. You can have a probe designed for it and come and tell you what FISH is, right? So FISH is a specific method to study a gene or chromosome. It just answers the specific question, does not screen for all abnormalities. We'll talk about details as we go ahead. Basically, you take the DNA, you denature it, you add the probe. Now, what is a probe? Probe is nothing but a nucleotide sequence which is complementary to your gene of interest, but this sequence has a fluorescent probe attached to it. So it will just go and home in on the gene which you want to study, and then you look under the microscope. What you see here in blue color, people, is the nucleus which is stained by DAPI, and this is an example of a 922 FISH probe. 9 is red, 22 is green. So normally you see 2 copies of the gene, so you'll see 2 copies of 9 and 2 copies of 22. So this is how it will look. And then if there is a translocation, you will see fusions. My teammates will go into further details about it. There are different designs of probes that can be used, and based on the question you are asking in terms of the clinical decision you need to make. Now, coming to the solid tumor fish. So, solid tumor fish is more technically demanding, right? You need tissue, you basically use formalin fixed paraffin embedded blocks. Now, you need optimal fixation, you need a good quality xylene, you need a good quality wax block. All that is very essential. You need a buffered formalin. Now, if pre-analytical variables are huge here. So we do get a lot of blobs from across the country, and if the processing is not good, you will not be able to do your fish properly. So basically, you need to process it. It's more technically demanding to ensure that your probe goes and binds on the gene of interest in the nuclei. So we do a lot of solid tumor fish. You'll come to know about as we move ahead. And as I said, there are a lot of pre-analytical recommendations. Now, coming to the terminologies, what are the parts of a chromosome? So, a chromosome has chromatids which join at the centromere. The short arm is P, the long arm is Q. And based on the position of the centromere, you have a metacentric, submetacentric, and acrocentric. And then, what we do is we follow an international system for cytogenetic nomenclature. So, whenever you look at the report, there will be an ISCN report. So this international system, actually lays down the whole guidelines, how a report should report, when should a comma come, when should a square bracket come, and so on. So in this, each chromosome is identified by chromosome number, arm symbol, region, and the band. So 1P31 means chromosome 1, short arm P for short arm, region 3, band 1. And then we have these kind of videograms we look back and refer to when we are reporting our cases to see whether any band is there, not there, there is a change in the banding pattern, and then we make a morphological diagnosis that yes, there is an abnormality on this chromosome. Now, whenever the reports are written, the number of cells are written in the square brackets, like here you can see. And then we have a whole lot of short forms we use. DEL is for deletion. You can have dicentric translocation. INV is inversion. We'll talk about it into more details. I'm just giving you a bird's eye view about different things. And what are the types of chromosomal abnormality? You can have numerical, they are aneuploid and polyploid. What do you mean by polyploid is? So as I said, you have diploid chromosomes, right? 46. So it's double of 23. Suppose you have 69 chromosome, then it is triploid. 92 chromosome, it is tetraploid. So it is basically the multiple of 23 is polyploid. Whereas aneuploidy is gain of certain chromosomes only. So you can have a monosomy, a trisomy, or tetrasomy. And in the structural abnormalities, you have translocations, inversions, insertions, deletions, you have ring chromosomes, isochromosomes, and extrastructural abnormal chromosomes. So as I said, aneuploidy, it can be 45 chromosomes, 49 chromosomes, 52 chromosomes, whereas polyploidy are multiples of 23. Structural, as I said, you have translocations, wherein two different chromosome exchange material. Inversion, where a part of a chromosome breaks and inverts itself. Insertion, a part of one chromosome goes and inserts itself into the other one. And then deletion, where pieces of chromosomes missing. Now, where are the applications? So the applications of cytogenetics are in constitutional abnormalities, children with dysmorphism or mental retardation, a couple coming with history of recurrence loss of pregnancies. So there you do karyotyping to see whether they have an abnormal chromosomes present. Patients with infertility. And then, of course, it has a huge range of application in cancer treatment. So hematological malignancies, and of course, in solid tumors. So this is an image of a couple, a karyotype from the lady, a couple who are finding it difficult to have a child. This image is from my friend Dr. Poonam, who works in Mumbai. So if you look, this patient had a 121 translocation. That was the reason that whenever the gametes were formed, and if it contained an abnormal one, any additional genomic material other than the sex chromosome and 21 is not viable with life. Your Edward and Patau syndrome, your trisomy 13 and 18 also die at the end of 1 year of their lives. Suppose you have an extra chromosome 1, it's not viable with life. That much extra genomic material will not work. Chromosome 21, yes, the children are born as Down syndrome. It's a very little amount of DNA on that, and the sex chromosomes. So you can have extra X chromosome where it is called as a super female. You can have Klinefelters. You can have a similarly loss of big chromosome is not viable with life. Right? Now this is another patient, and you can see it has a 121 translocation again. Now, can you tell me which of the following karyotypes are not compatible with life? 47XY plus 21, 47XXX, 47XXY, and 47XY plus 2.
47 triple.
Okay.
B.
47 XY plus 2.
Yeah, so that is the only right answer. The first three were all wrong. As I said, only extra chromosome 21 and sex chromosome is viable with life, right? So if a zygote gets an additional chromosome 2 there will be spontaneous abortion. It will die. That 2 is a big chromosome. That much genomic material will not work at all, right? The sex chromosomes, because in the X chromosomes, there are a lot of genes, but as you know that one copy of the chromosome is usually undergoes lionization. So all the genes on the sex chromosomes are not expressed. So that is viable with life. And Y chromosome is a very small chromosome, poor thing. Even if it is lost, it only leads to sexual ambiguity, but it is viable with life. Yeah. So having finished the introduction, we will go into more details about fish. But before that, let me just show you some videos to give you an idea of what is done. Can you see the video?
No, sir.
No, sir.
Okay.
Not yet.
Okay. I'll just stop sharing and share again. Let me see if that helps. Now?
Not yet. Yeah, yeah. You can see,
Yes, sir.
Yeah.
Yes, sir.
So what you see here is what we are doing setting up. So basically you can see the HAPA. Okay, let me go into, I hope you can see the arrow. Anyways,
Yes.
you can see the green top heparin vacuoteners, and these are the culture tubes. So what Pranay here is doing is, you see, is looking at the bone marrow samples of the same patient. Now there's one more person here, Tuhin, who is actually telling him what are the clinical details. Is it a leukemia? Is it an MDS? And then we are actually cross-checking the name of the patient number, because remember, a mix-up here is disastrous, because what will happen is then, see, unlike in histopathology, if there is a mix-up between a testicular biopsy and a brain biopsy, you can of course go back and say yes, there was an error, there was a mix-up. But here everybody's chromosomes look the same, so you just cannot mix up. So at every point you have to see to it that the right sample is identified, and we only take one sample in the tube in our hood at a time. Now, why are we using a laminar hood? Because as I said, we'll be putting culture. It's 37 degrees, it's 90% humidity. That is fantastic for fungi to grow. So our culture media also has antibiotics and antifungals. So what you see here, this beautiful, what color would you call it? Purple? Orangish, purplish color, guys are really bad in colors. So whatever this, so this is the media, and then you just look at the counts. So what we do is, you must understand one thing. See, you are creating an artificial atmosphere here for the cells to divide in a tube, right? And this is 5 ml of media we are using. Okay, so generally you need to have about 1.5 million cells per ml of media. So what we do is, we first calculate the amount of number of cells in the marrow, yeah, the WBCs, and then based on that we calculate how much volume of bone marrow has to go. So for example, if it is an aplastic anemia, then you need to add more marrow. If it is a leukemia and there are a lot of cells, you need to take a little amount of marrow and put it, because if there are too many cells, this is a limited space. They will kill each other for the nutrients. They are fighting here for nutrients. They are fighting here for oxygen. They are fighting for everything, right? So you need to provide them the right space for that. And now what he's doing is, he's adding colcimate. Now this colcimate will arrest any cell that undergoes division, right? So I'll just forward it a little. So he adds the colcimate here. And then you go into the CO2 incubator, right? So these are the carbon dioxide. As I said, there's 37 degrees temperature, 5% carbon dioxide. And these are the tubes which have already been placed. They are put in lying down position. And we keep the cap a little loose because, the carbon dioxide exchange takes place. So the tubes are put overnight into these carbon dioxide incubators, right? So that the cells keep dividing. And as I said, every time a cell undergoes division, it is arrested. Now, next day morning, we do what is called as harvesting. So this is harvesting. We have taken the tubes out. Next day morning, we have centrifuged them. And what you can see here, the arrow is the pellet, and this is the media which is left. So we remove the supernatant, yeah, and then we are adding the hypotonic solution. Why hypotonic solution? The hypotonic solution will go inside the cell, and the cells will start ballooning up, and the chromosomes will start separating, right? So you tap that way, you add the hypotonic solution drop by drop from the sides of the tube. You don't want to put it bang on the head. So it's like in the, I don't know how cold it is in your part of the countries where you are. So you go to take a shower and the cold water falls on your head, right? It's a shock. So we don't want to shock the cells. So what we do is we put it gradually, and then we move ahead and tap. And after that, we after putting the initial part, we put it in 37 degrees. Give it a 5 minute, 10 minute here, and then after that we add fixatives, and we go on to do that until you get a white pellet, right? So what you see here is something which has been treated with hypotonic solution, treated with fixative, and now we have a white color pellet. The RBCs have all gone away now, right? They have all got lysed by as adding the fixative, which is nothing but the carnoise fixative, which is glacial acetic acid and alcohol. Your glacial acetic acid will just destroy all your RBCs. You give multiple washes, and then you have this. The next step is making slides. So just to summarize, we took the bone marrow, we put it for overnight culture, we added hypotonic solution, we gave it fixative washers, and now we have a pellet of cells, and then we are harvesting, right? So the slides here are kept in chilled ethanol. So I'm sure all of you know that ice goes very well with alcohol. So you take chilled alcohol, you take some chilled water, dip your slides into it, and then what she's going to do now is she is going to drop the cells with the metaphases on the slide. There she drops it, right? You can say she is dropping it, right? Now you must understand, that slide making is, so one thing about cytogenetics is every case is different, so uh, we have to dry the slides, and as the slides dry, the chromosomes start spreading out by the interaction of the fixative and the chilled alcohol. So what you have to do is based on every case, you can change your drying time. You have to keep changing your drying time, and it also depends in which part of the country you are. Slides making is difficult where it is if it is very humid, right? So you need to have a controlled humidity, you need to have controlled temperature. And here she's making the slides. You can see she's drying with her hand. She has a stopwatch in front of her, that is Shipra. So Shipra is a part of our team. She is a pure vegetarian but serves us the best fish in the country, right? She produces beautiful fish slides, both solid and liquid tumor, and they're just beautiful. I mean, when you peep into the microscope, it's an amazing world below there. So now she is drying the slides for you. And then we look in the phase contrast microscope. She will look at it and see whether the metaphases have dried properly or no. It is very important. Each and every step in cytogenetics, a small deviation can completely change the quality of work. So chalta hai, yahan par nahi chalta hai. So in cytogenetics, the chalta hai attitude doesn't work. You just have to be very meticulous. It's a lot of meticulous repetitive work, and it should, and believe me, when you see the results, you see the cases my fellows will show you, it does excite us every day to work here. Now I'm opening another video. Just let me know whether you can see the video or not. Can you see the video?
Not yet.
Yes.
No, yes.
Yeah. So this is Pranay. He's the fashion icon of our department, right? Now what we have done is, you see the, okay, let me pause it. So what you see here is a microscope. These are the slides from a patient. We have captured the metaphases, okay? Let me go back. Yeah, so this is Pranay Gurung for us, yeah? And you see this microscope, the slides, the images have been, so this is the image of a metaphase which has been stained by James Sa. Now he's using a software to analyze it. So what he's doing is he's just using some settings to make the chromosome look more beautiful. The banding, you can see this is how a metaphase looks, right? And now, see, the chromosomes will be stuck to each other. So you need to identify the chromosomes, and then you need to separate them out, right? Now, for example, here there is an overlap, right? So you need to see, if you don't, so actually, we first look under the microscope and identify the chromosomes, right? And just there itself, we say, what is the diagnosis, right? So that's called I-karyotyping. And so he separated this chromosome. You see, these two chromosomes were hugging each other. He just removed them, and separated both of them, right? And then he sees, okay, are they all separated well? And after that, he will look at the banding and he will go on to arrange them. So there is no software in the world so far that people have worked at AI to see that the chromosomes can be identified by the machine. So once there was this big CEO of an IT company visiting our lab, and he saw this and he told his subordinates, "Get two guys from IIT on a PhD project. Let them develop an AI to identify the chromosomes. We'll do it in 3 months." I said, okay, you take the images. It's been more than 4 years, they're still struggling, right? So that makes us, we are still in jobs. So, AI is, at least for now, not replacing us. Yeah. So once these chromosomes have been arranged, then we look at the banding patterns. We look at least 20 metaphases in every patient, and we look, are there any abnormalities? Of course, we correlate with the bone marrow morphology. We correlate with the flow cytometry findings, and with all of that data, we sit and analyze and then give our reports, right? So I'm going back to the presentations. Can you see the PowerPoint?
Yes, sir.
Yes, sir.
Yes. So I'd like to invite my colleague Samipa now. Samipa, all yours.
So before going ahead, we have few questions in the chat
Okay, you
box.
want me to take them now? Sure.
Yes. So the first question was asked by Dr. Sundar. The question is what to do if we cannot take bone marrow aspirate specimen for cytogenetics in cases where there is packed marrow or there is myelofibrosis. Can we do cytogenetics in any other way?
Okay. So there are two aspects to it. For example, you have a packed marrow of a leukemia, and you are not able to do MRO, or a patient comes in late in the evening with very high counts. The treatment has to be started immediately, and you can't wait for the marrow to be done. The peripheral blood has given you the diagnosis. So if your peripheral blood has a good number of blasts, has a good WBC count, of course you can take the peripheral blood and put it for karyotyping. But please remember that the number of metaphases you get can be fewer. The chances of failure can be higher. So in our lab, we have 90% success in getting metaphases, which is as good as anywhere in the world, rather, I would say. And we have in myeloid leukemias, at least 60-70% of our patients have abnormalities on karyotype, and similarly with lymphoid leukemias, right? So, in order to you must understand, when the blasts are in the marrow, they have those addition molecules with which they interact with the stroma, and they have a higher proliferative potential. Once they've gone into the peripheral blood, they would have lost those their proliferative capacity would have come down a little bit. But if there is no choice, we have to do that. Now, if you have a patient with myelofibrosis, you can't get a marrow at all. And if you get a hemodilute marrow, we will still go ahead and process the marrow. If it is a failure, it is a failure. We can't do much about it because even the peripheral blood will not have enough cells. One of the biggest problems which I have seen when I get cases referred across the country, people will do PHA stimulation in such patients and give a karyotype. It is ridiculous because the PHA will only stimulate your T cells. You are not getting metaphases from your myeloid cells. Okay, so there it will be a problem, right? That is not the way. And I've seen patients with myeloid malignancies getting a peripheral blood PHA stimulated karyotype and the report being given as normal karyotype. Of course, it will be normal. You're looking at the T lymphocytes there. So these are the possibilities, and we have seen sometimes with hemodilute marrow with very few cells also when they have we have pampered these cells in our media with a lot of nutrients, we have got good number of metaphases and we have been able to give diagnosis. So it's a case to case basis.
So there's another question. I think Dr. Soumyadeep has put it, that what is the significance of the blank area which is marked as MAR after chromosome number 5?
Okay. So as I said, each chromosome is identified by its banding pattern. Okay. Sometimes the rearrangements are so complex that you don't understand what this chromosome is, or from where these material is coming from, which chromosome. So they are called markers. So the marker simply means I don't know, right? So sometimes in a lot of complex karyotypes, you will get small structures of chromosomes which you are not sure it's coming from which chromosome. So those are called markers. Sometimes based on our morphological evidence, like how you do histopathology and then put IHC. Looking at that morphology, we do put sometimes specific fish probes to identify and what is abnormality. And sometimes, yes, we are bang on, sometimes we are not. And we just put it as marker, which clearly means that there is a structural abnormality which is too complex for a normal for us to identify from where the abnormality is coming.
Thank you, sir.
Yeah, thank you, sir.
That's it?
Yes,
There's another one. They are just asking that can we use bone marrow biopsy in case of dry tap by Dr. Khushbu?
Yeah, so Khushbu, as I told you, we need live cells for karyotyping, right? So, but a bone marrow biopsy will not have live cells, right? It'll go, it'll get fixated, you will use formalin fixed paraffin embedded tissue, you would have undergone decal, the cells are dead, so you cannot do karyotyping. The next question you may ask is, can you fish a bone marrow biopsy? Now, you must understand that decalcification, with those acid treatments, denatures the DNA, and hence the fish may not work. So, EDTA based decalcification or a mild decalcification is good in order to see to it that your decal is mild so that you can use fish probes in a bone marrow biopsy. Now, also you must understand, if you look at this image you are seeing, right, you just see these blue cells. Now, if your bone marrow is not completely replaced by blast or if there is patchy infiltration, it is very difficult for us to understand whether this nuclei is erythroblast or this nuclei is a myeloblast. We can't identify that at all. So those are the challenges with bone marrow biopsy fishing. But yes, in patients we have fished. Sometimes it works pretty well. Sometimes it doesn't because it all depends from how the marrow was processed, how it was decalcified, and these are the challenges we face on a day-to-day basis. Yeah.
So there is one more question. Why CO2 incubator is used?
Okay. So as I told you, the bone marrow cells are proliferating, right? Now, when you put them into the media, they will produce what are metabolites, and one of the metabolites is the what is called HOC3, the carbonic acid. So, in order to neutralize and to prevent excessive acidity being formed, acidic milieu being formed within the closed space, the carbon dioxide is exchanged, acts with HOC3 minus, and produces carbon dioxide and water, which is released. So, in order to maintain the pH of the milieu, because see, there's all cells are dividing. You've given them everything to eat and enjoy. There's a party going on in there, right? So when a party is going on in there, you need to maintain the pH, and that is why the CO2 actually kind of helps you to get. Now you may say, well, I don't have a carbon dioxide incubator. I will use a normal incubator. You can still get metaphysics, but the chances of failure increase.
Done with the questions, sir. We can now proceed further.
Yeah. So over to Samipa.
Thank you, sir. Thank you, sir.
So, good afternoon, everyone. So, and I will be talking about the basic procedure or the basic protocol of both liquid
and solid fluorescence in situ hybridization, that is FISH. So before starting, we all should know that what is FISH? So it is a cytogenetic technique which is used to visualize specific DNA sequences of the genome under the fluorescent microscope. Now, a FISH can pick up minute gene rearrangements by using either fusion probe or a break apart probe. It can also pick up the copy number changes using probes for amplification and that for deletion. Now let's see the principle of this technique. So the basic elements for FISH are probe DNA and the target sequence. Now the probe DNA are first labeled with fluorophores. Then both the probe DNA and the target sequence are allowed to get denatured. Once denatured, this probe DNA will find its way into the genome and will get hybridized to the complementary base pairs. Now, these fluorophores will then emit signals under the fluorescent microscope and will help us to visualize the specific or the gene of interest which we are seeing for. So after this brief intro, so there's a question for you all. So what is the false statement regarding FISH?
A.
It
It analyzes the
analyzes the
whole
whole
A, ma'am. Exactly. So FISH is a technique where we are visualizing or seeing specific sequences, not the whole gene.
Yes.
So let's see on what kind of samples we can perform FISH. The liquid FISH or hematological FISH, we do it from peripheral blood as well as from bone marrow samples. Preferably, we collect the samples in heparin vials. And for solid FISH, we can perform it from formalin-fixed paraffin-embedded tissues, though that is the FFP blocks from the cell blocks or the cytology smears. We can also do solid FISH from the imprint smears as well. Now let's see the basic workflow of the liquid FISH. So liquid FISH is a 2-day procedure. On day 1, first we prepare the slides. We prepare the slides from the fixed pellets and then we mark certain areas on the slides. Here you can see I have marked 3 areas. So based on the cell density on the slide, we mark the area so that we can add the probes on those particular areas only. Next, we treat the slides with a buffer. So the buffer we use here is sodium saline citrate, that is 2x SSC, which maintains a pH of 7.0, followed by a dehydration with a series of alcohol wash, and then we add a magic potion, that is the probe. We cover the area with a cover slip and then we put a temporary seal. We use rubber cement for this temporary sealing purpose. Then we put the slide in a thermobrite. So this thermobrite is like a thermal cycler. So here first the temperature will rise to 73 degrees and will maintain it for 5 minutes. So here the probe as well as the target sequence will undergo denaturation, which is followed by hybridization, where the temperature will come down to 37 degrees Celsius and it will maintain it for about 12 hours. So basically what we do is we keep it overnight for hybridization. So from here, we jump on to day 2. So on day 2, what we will do is post-hybridization wash or the stringency wash. So the first thing what we do on day 2 is we remove the rubber cement or the temporary seal which we had put on the cover slip. Then we put the slide or we dip the slide in 2x SSC again. So this 2x SSC will ensure that the cover slip will come automatically. We will not put any force to take it off. Then we wash the slides with 2 changes of Igpal. Now what is Igpal? It is nothing but a liquid detergent which will wash away the unbound probes. Followed this, we do the counter staining. We use DAPI for counter staining, which will stain the nucleus. And then we go for analysis. So this is how a liquid FISH shows under microscope. The interpretation part will be discussed by Mr. Arun in the following lectures. Next, I will move on to the solid FISH procedure. So solid FISH is a 3-day procedure. On day 1, first we ask the histopath people to get us the required sections from the required FFP block. So we take HE slides as well as unstained slides. The tissue thickness must be as thin as 3 micron, and the slides which are used for solid FISH are coated with poly-L-lysine. Now you may ask that why this coating is required. Because both the glass slides and the tissue sections are negatively charged. The glass slides will have free hydroxyl ions and the tissue section will have free carboxylate ions. So both these are negatively charged. This will repel each other. Thus, our tissue section may wash away while we are doing the procedure. So polyl contains a lot of ammonium cations. Now these cations will form ionic bonds with both hydroxyl ions in the glass as well as the carboxylate ions which is present on the tissue. Thus, this will help in adhesion of the tissue on the slide. So after getting the unstained slides, what we do is called baking. We bake the slides at 56 degrees Celsius overnight. With this, we move on to day 2 procedure of solid fish. So first we do deparaffinization because on the FFP block there are a lot of paraffin wax. So we need to remove this wax. So for this we put the slide in xylene. We treat it with a series of alcohol wash for rehydration after the deparaffinization, and then we do the heat pre-treatment using 0.2 normal HCl, which will reduce the autofluorescence, followed by digestion of the methylene bridges by sodium thiocyanate. Now this sodium thiocyanate treatment will actually make way for the probe to enter the cell. Next, we do the enzyme treatment using pepsin, which will digest a lot of proteins which are present on a solid tissue section, followed by dehydration with a series of alcohol wash, and again then we will add a magic potion, that is the probe. We'll put a cover slip so that the probe which we have added is covered, and we put a temporary seal using the rubber cement. We put the slide in the thermobrite, which is the thermal cycler, which is a type of thermal cycler. So here for the first 5 minutes, the temperature will go to 73 degrees Celsius, which will help in denaturation, followed by hybridization at 37 degrees Celsius, and will keep this slide for hybridization overnight. With this, we go to the day 3 procedure of the solid fish, where we will do the post hybridization wash. We will remove the rubber cement or the temporary seal, and then we will dip the slides in 2-XSSC so that the cover slip will come off automatically. Then we wash the slides with 2 concentrations or the 2 changes of Igpal, which is a liquid detergent, which will wash away the unbound probes and will clear the background. Then we stain our slides with DAPI, which is the nuclear stain, and go for analysis. So this is how the solid fish looks like under microscope. Again, the interpretation and the probe design and everything will be explained by Mr. Arun in the next lectures. So thank you. This is all about the basic procedure of liquid and solid fish.
Thank you.
Any questions you can ask?
There are no questions in the chat right now, so I think we can move forward. Or if students want to ask anything,
No.
We may proceed further now.
Yeah. Thank you.
Thank you so much.
Good afternoon, everyone.
Good afternoon.
In next 10 minutes, I'll be talking about the various fish probes, its designs and the types. So this will help you to select what kind of probe has to be used for the particular abnormality. So now I hope everyone will be knowing the basics of fish and the principle. So this I'll quickly summarize. So this is our target DNA or the patient sample, and this is the probe DNA. Both are double-stranded now. So now the probe DNA is labeled with the fluorescent dye. Next step is the denaturation, where both the target DNA and the probe will become denatured and become single strands. Now next step is for hybridization. At lower temperature, these single strands, the probe DNA will get more affinity to bind back to the target DNA. On the next day, under fluorescent microscope, we still see the signals. So this is how it works. Now coming to the selection of various fish probes. currently in the market there are various companies who supplies the common probes. You can select from these the various companies based on the price, the quality of the signals and all. So now there are some of the companies who can give you custom-made probes which are not available in their catalog and all. Even if you have the good setup, like assertive conditions, you can make the probes in your department itself, in your lab itself, but it is again hectic. Coming to the various types of probes, so these are the common probes. First one is syndrome enumeration probe or CEP probe. Next one is locus specific probe or LSA probe. Third one is telomeric probe. Last one is whole chromosome paint or the WCP. First, I'll talk about CEP. Centromere is the primary constitution. The centromere is determined the chromosome. So the CEP probes will bind to the centromere of a chromosome, and this will show whether the chromosome is present or not. That will help you there. Next is locus specific probes or LSA probes. So as the name suggests, it particularly binds to a locus of interest. It can be a gene or it can be a region of interest. Next one is telomeric probe. It binds to the both telomeres of the chromosome. So ideally, a chromosome should have two telomeric signals. And the last one is whole chromosome paint or WCP. As the name says, the whole chromosome gets painted. The first two, that is CEP and LSA probes, are more used in a diagnosis setup, whereas the telomeric probe and WCP probes are more used in a research mode. So these are the common examples for these various probes. First one is CEP probe. So here I'm showing the example of CPXY. So in a case of post-transplant where you have to check for the graft success, you can use a CPXY probe. That is a fish-based chimerism, provided there should be a mismatch there should be a different sex between donor and recipient. Here on the left side, you can see green and red. So green is chromosome Y, whereas the red is chromosome X. So this is XY, and on the right hand side, this is XX. So usually chimerism reports 1,000 cells are red, and like the report will contain a number of donor cells and recipients. So this is a example for CPXY probes. There are more other probes which are using CPXY. The next one is telomere probe. So these telomere sequence, the telomeric probe will bind to the specific to the telomere sequence. This is specific to a 300 kb locus at the end of a specific chromosome. Third one is whole chromosome painting. So in a WCP, the multiple probes are labeled. So this helps to hybridize along the whole length of the chromosome. On the left hand side, you see two chromosomes. This is a green color. This is a chromosome 2. The whole chromosome got painted with green color. And here the chromosome 5 is there. Its whole chromosome is painted with red color. So on the right hand side, the counterparts which are abnormal, you can see a color change here. So here whole green is there, but here there is a red color is there. Same way on chromosome 5 also you can see green dot here. So there is a reciprocal transfer between these two chromosomes. So WCP probes can be used to pick up translocations, but it can't pick up deletions, smaller deletions and inversions. Even duplication also it can't be picked up. The last one is LSA probe or the locus specific probes. So as the name suggests, it is used to pick up any deletions or any translocation or even amplification. As I told earlier, LSA and CP probes are the commonly used in a routine diagnosis setup. Now coming to the various probe design, these are the commonest ones: fusion probes, break apart, and enumeration probe. So before going to the detailed things, so I'll show you an example of a translocation. So this is a case of a CML. So you have a chromosome translocation 922. So on the right side you have the abnormal chromosome. This is abnormal chromosome. This is abnormal chromosome. So what is happening is from the 9, that is at 3-4 region. This is the region of the particular this is a locus. So from 3-4, a part of the 9 is going. That is ABL. It is going to the 22. So there is a 1.2, that is the locus of BCR. So from that, since it is a reciprocal translocation, this is going to 9. So this is how it is happening. So this is a reciprocal translocation. So now coming to the first example, fusion probe. So again, I'm using BCR label as an example. So BCR is labeled as green, ABL is labeled as red. So the color coding, it varies from company to company. It is not necessarily always BCR will be green, but most of the companies, they use BCR as green and ABL as red. So now the first interface cells you are seeing, 2 green, 2 red. So this indicates there is no translocation. There is no exchange between any segment. Now the second image, if you see, the pattern what you are seeing is called 2F, 1 green, 1 red. Or it is 2 fusion, 1 green, 1 red. So various labs use different terminology. So commonly it is called as 2F or 2 fusion. So what is happening is here. So every chromosome has like every gene has two alleles. So there are two ABLs and two BCS. So this is a normal allele, normal ABL, red color here you can see, and on the right hand side you can see another normal BCR. So what is happening is there is a break happening at ABL1, and the part of ABL, that is a red color, it is going to the 22 where you have the BCR. Since it is a reciprocal translocation, a part of the BCR goes to ABL. So finally, the resulting chromosome on 22, the chimeric gene is called BCR ABL or the Philadelphia chromosome, and on 9 you have ABL and BCR, which is not pathogenic. So this is the explanation: 2 fusion, 1 green, 1 red. So this is the pattern for most of the fusion probes. So this is an image from the article published in IJPM. So from my user and team, they have published in IJPM, like it is in a CMC paper. This is one of the largest study on desirable fusion patterns in CML. So we have seen 2 red, 2 green, which is a negative thing. Second is 2 fusion, 1 red, 1 green, that also we have seen. So these are the various atypical signal patterns, which I'm not going to explain now. So people can go through this paper, so you will come to know the details of the various probe designs for the type of signals. So these are the other common probes which are used as fusion probes: 1517 of Amelara, 414, 1114, 1416. These are in myeloma. 920 we have seen, 821. There are many more fusion probes. Now coming to the break-apart probes. So previously we have seen fusion probe where you have the two fixed partners. So now if you see this image, this is a partial karyogram. So there are multiple karyotype images you are seeing. So these all are from our lab. So if you see what is common is that, so I will show you. First one is 211, second is 411, 611, 911. So these, everywhere what you are seeing is chromosome 11 is common. So chromosome 11 houses KMT2A gene or the MLL gene, which is like rearranged in some of the acute loops. So here if you see on the right side, you can see KMT2A, these are the. So here on the right side, you can see the different partners. So in AL itself, there are 100 plus partners. So if you go by a fusion probe design, so you have to design 100 plus partners. So that is not technically possible. You don't have time, you don't have money. So the best design to pick up rearrangement of a gene which is having multiple partners is a break-apart probe. Yeah, this is a break-apart probe. So you can see the ideogram where the signals are there. So previously we have seen BCR ABL. So BCR and ABL are on the different chromosomes. So the different signals are there. So now what you are seeing is that red and green are fused together to a single gene. So now what we are explaining is MLL gene or KMT2A gene. So every gene has its 5' end and a 3' end. On the 5' you can have a red color, and the 3' you have a green color. So these are fused together and you are seeing a fusion here. So on a negative cell, so this is a negative cell, we have two fusion, there is no rearrangement. So whenever there is a rearrangement, what happens is, so this is the terminal part. So this green will go to the partner chromosome. So if you see here, so this is the abnormal chromosome, this is the normal chromosome. So the fusion will be on the normal level, and here instead of fusion, the signals will be break apart. So here green will stay and the red will go to the partner chromosome. So this is chromosome, this is a transcription 411, one of the abnormalities in the childhood ALL. So this is a metaphase showing rearrangement. Here this is a normal or intact MLL gene. So here green and red are separate. So this is how we interpret a break apart probe. Always keep in mind the distance between two signals has to be double the size of the signal. Then only we'll call it as positive. So coming to the examples of break apart. So most of the cancer has various genes which have multiple partners. So the best strategy to pick up the rearrangement of these type of genes is fish using a break apart probe. So there are almost 50 plus probes which we have as break apart probes. So both in solid tumor and the IMAX. Now coming to enumeration probe, these enumeration probes are used to detect copy number variations. So this copy number can be either gain or amplification, or it can be loss or deletion. So in this scenario, what we have first we'll have a CP probe which will act as a control, and there will be an LSA probe which acts as which is a gene of interest. So on the left hand side, these are the examples for gain. So HER2, NMIC, MET, these are the examples. On the right side, you have deletion. So it can be TP53, ATM, RBU. These are the common ones. So now first one is the first one is a HER2 probe. So this is the kit insert about the probe design. So this is a CP probe. So you have a centromere which has green color, and on the Q, that is a long arm of chromosome 17, you have HER2 gene. So this is a green, this is red. So ideally there should be two green, two red, representing two chromosomes 17, two chromosomes of HER2. So these are the different images. So these more details will be shared in the coming lecture. Now coming to the copy number losses, the best example is TP53. Again, chromosome 17 is an example. So you have the CP probe of 17, which has green color, and the TP53 gene is on the short arm of chromosome 17. It is labeled with the spectrum orange color. So in a normal scenario where there is no deletion, you will get two green, two red. So this two green, two red indicates there are two copies of chromosome 17 and you have two copies of intact TP53 gene. So now this is negative for a deletion. It's only negative for deletion. Now next is the two green, one red pattern. So here you have two copies of chromosome 17, but only one copy of red. So that indicates there is a deletion. So now come to the last state, tricolor probe. In cytogenetics, fish is not restricted to two colors, that is green and red. So we have other colors like aqua also. So we have some probes called PD5 alpha rearrangement, MEF2D. These all things we have a tricolor probe. Thank you. Any questions?
Any questions?
Yes, sir. There's a question that by Krishna Mohan Singh, that can we design and synthesize different types of fish probes?
Yeah, that is possible. So if you have a good lab which are aseptic conditions, you can design. But again, it is more hectic. So better, most of the probes are now, the common probes are available across the different companies. Or some of the probes you can ask, which are not, if it's not available, you can ask some of the company to make a custom made probes. So it is possible that way.
It is not easy to make a probe. There is a lot of cross hybridization, a lot of QCs. So I think why reinvent the wheel when there are experts who can do this for you? So there are companies where you want a specific probe for yourself for a research question, they will develop it for you.
Okay. The other question is from Rahul Vastogra. Is fish can be used to detect new anomaly or can only be used to confirm any abnormality that we are suspecting?
No. Like if you these are this you can't pick up. The fish is used mainly to detect the known things. In the case of first, if you don't know the details of the thing like abnormality, you can't use fish to pick up those abnormalities.
Last question. Is there any difference in the diagnostic sensitivity and specificity between liquid and solid fish? Like, for example, a fresh bone marrow aspirate versus a formalin fixed bone marrow biopsy.
See, you have to understand one thing, both are completely different techniques, right? And in terms of sensitivity and specificity, it is calculated for each single probe. So we haven't kept a talk for that to go into details. But for every single probe when you get, even if you're buying it from the best company, you actually calculate the sensitivity and specificity. You do run certain normal controls, you do CV calculation, and then you calculate the own cutoff for a specific probe. Now, in terms of using the same probe on a bone marrow biopsy. Now, you must understand that most of these genetic abnormalities at diagnosis are founding abnormalities, or they are the driver genomic events that has caused the leukemia to happen. So at diagnosis, it shouldn't be an issue. Even if you're looking at a solid tumor and say a CML biopsy will show everything with 920, right? So only problem is when you're dealing with post-treatment or partially treated samples. But anyways, for most of these patients, post-treatment follow-up, cytogenetics is not the best technique to look at that. So yes, you do have to calculate sensitivity and specificity, and yes, there is a difference how you interpret them in liquid and solid tumors.
Okay, thank you, sir.
So next, we have a fellow Dr. Aishwarya, who is going to talk about. So she's going to share our experience in hematological malignancies.
Good afternoon, everyone. So today I'll be talking about the cytogenetic aspect in hematological malignancies. So why do we do cytogenetics in hematological malignancies? So it's basically in terms of diagnosis, follow-up, for risk stratification, characterize, and for particular directing treatments. So how do you write a karyotype in general? So first we follow the ISCN 2020 guidelines, which is the latest guidelines. Then we have the number of chromosomes. Then we have the sex chromosomes. And this, the number in the box bracket depends on the number of metaphases you've analyzed. So in this, we have seen a normal karyotype in 20 metaphases. So similarly here, so this is the total number of chromosomes. This is the sex chromosomes. This is the arrangement abnormality. So they can be, we can have deletions, translocations, inversions, insertions. So here we'll have the symbol for the particular abnormality that we have detected. This is the chromosome on which the abnormality we detect. P and Q symbolizes the arm. So whether it's the short arm, that is P, the long arm, that is Q, and 3-5, that is the band, and the subband, that is the segment where it is involved. So here we have a deletion, example of a deletion, and here we have an example of a translocation. So this translocation that we see is seen in 19 metaphases, and one metaphase that we analyzed was a normal metaphase. So why is it important to know this? Because whenever you see any abnormality, you have to have it established as a clone. To call it as an abnormal karyotype, you have to establish it as a clone. So clone is defined as the cell population derived from a progenitor. So as earlier I showed, this is the number of metaphases that we calculate. This abnormality has to be present in the number of metaphases that we've analyzed. Like for example here, this is a female karyotype where there is presence of a translocation 821, which is seen in 20 metaphases. So why is it important? Because depending upon the type of abnormality that you see, you have to establish it as a clone. There are a minimal number of metaphases where it should be analyzed. So for a trisomy, it should be at least present in 2 metaphases. For a monosomy, in at least 3 metaphases. For any structural abnormality, that is the translocation, the inversions, the deletion patterns, it should be seen in at least 2 metaphases. So the number of cells that constitute a given clone is the one that we give in the square bracket. So can anyone tell me a condition in which there is only one copy of a homologous chromosome pair? What do we call it?
Monosome. Monosome.
Yeah, correct. So like for example, now here we have a metaphase showing monosomy 7. But now is it clonal and how many metaphases is showing it? So as I described earlier, for a monosomy to be present, it should be at least seen in 3 metaphases to call it a clone. So as we all know, monosomy 7 is a poor risk factor in terms of in myeloid neoplasms, right?
MDS.
Yeah, so this is how we generally write, where we give the specimen type, the banding resolution, that is at which bands per haploid set it is seen, the number of metaphases it is counted, the number of metaphases analyzed, and the total number of chromosomes. So here the mistake that they have given is that they have not mentioned that in how many metaphases they have seen a monosomy 7, which is extremely important in terms of the clinician. So do you think this is the correct way of writing monosomy? Okay. So here they are saying that they have seen a monosomy 7 in 7 metaphases. So as I said earlier, to call it a clone, it has to be at least seen in 3 metaphases, correct? So here it is seen in 7 metaphases, so this is an acceptable statement, okay? So similarly here, now here monosomy 7 is only seen in 1 metaphase, so the clonality is not established. So we cannot say that monosomy 7 is present. It can be also a random loss. So as the videos which were shown earlier, when we analyze, sometimes a small chromosome can be lost from the edge if it is seen. So that is just a random loss. So to call it a clone, it has to have been seen in a specific number of metaphases. So the next step in such cases, we can do a FISH for monosomy 7. So coming to translocations. So as we know, translocation 922, translocation 821, these are the commonly known translocations that we see. So there are two types of translocations that are seen, balanced and unbalanced. So in case of a balanced translocation, for example, this chromosome A and B, these break at a particular breakpoint and the genetic material is exchanged. So there is no gain or loss of any genomic material. So this is an example of a balanced translocation. Whereas in terms of an unbalanced translocation, when we have these two chromosomes which are the parent one, so we have genetic exchange of material, and this is the translocation that we get. So this here there is no equal exchange as seen. There is no gain of genetic material by this chromosome. It's only seen in this chromosome. So this is an unbalanced translocation where you can have loss or gain of the genomic material. So how do we write a translocation? We write it as a symbol T, and we describe the smaller chromosome is always written earlier, followed by the next chromosome followed. And in case of an unbalanced translocation, we use a term known as derivative, which I'll be explaining further. So what are the effects of these translocations? There's deregulation of the genes by juxtaposition of the strong enhancers in cases of IGH c-MEX translocations, or you can have formation of chimeric fusion genes for hematopoietic transcription factors, epigenetic modifiers, cytokine receptors, and the tyrosine kinases in terms of 922. So do we always need a blast? Can anyone answer this to say that a particular abnormality is seen?
No.
In case of leukemias, yeah.
Yes, not always blast is required. Recurrent cytogenetic abnormality can be stem as a acute leukemia.
Correct. So we had this case of a 14-year-old female who had presented with cramping muscular pain on the upper and lower limbs, and she had on and off weakness for a month with skin bleeds lasting for about 3 days. And on examination, she had pallor and no active bleeding. So her aspirate showed marked hemophagocytosis and dysgranulocytosis features, and her blast percentage that we calculated was around 2%. So on IPT there was no gating. The 70% cells were gated in the granulocytic window, and there were no gating of blast cells. They all showed loss of CD10 and CD15, and there was an aberrant expression of CD56 which was seen. So there was just dysplasia and no increase in blasts. So on karyotype, what happened is we found this. Can anyone guess what translocation this is?
It's 821 translocation.
Yeah.
21.
Deletion
Correct.
of.
Along with this is an interstitial deletion of the long arm of chromosome 9.
Okay.
So 821 was an AML which was characterized as a separate entity in WHO 1998. So basically, if there is presence of this abnormality, you do not need to check the blast percentage. You can directly call it an AML with an RUNX1, RUNX1T1 translocation. And the same goes for cases of inversion 16, that is the core binding factor beta with MYH11 fusions and your PML-RARA translocation. So next is a case of a 40-year-old female who presented with AML, and she had an aberrant CD19 expression. So can anyone guess the abnormality?
So translocation 821.
Correct. So we even had a FISH for this patient, and it was positive for RUNX1, RUNX1T1 translocation, showing a 2 fusion, 1 red, 1 green pattern. So this was a dual color, dual fusion probe that we had used. So why is it important to sometimes FISH it? So 5% of these 821 abnormalities can be cryptic on a karyotype. So if you have an aberrant 19 and you're suspicious of 821 on the marrow features, so you can always FISH it and confirm it. Can anyone enumerate me some marrow features which can be seen in, you can suspect a 821?
Sorry. It's M2 type, maybe.
Yeah. So basically, you can have large blasts with basophilic cytoplasm with numerous azurophilic granules, a salmon pink-colored cytoplasm. Few of these blasts can show these pseudo-Chediac Higashi granules. You can have sometimes few ore rods which can be seen, aberrant CD19 which can be seen on IPT, and there will be dysplasia in at least 2 cell lines which can be seen on the aspirate. Along with it, sometimes on cardio, if you have these associated secondary abnormalities, you can also suspect for an A21 translocation. Especially if you have trisomy 8, if you have loss of the sex chromosomes or the deletion 7q or a 9q. So these are, according to the newest WHO version, so these are the cytogenetically defined abnormalities which are seen. So in cases of PML RARA, 821 and core binding factor beta, we don't have to have a particular blast percentage. If these abnormalities are present on karyotype, you can straight away call them AML. In cases of DEC NUC214, you will have basophilia, which will be seen on the marrow. In cases of RBM15, which is basically translocation 122, which is seen in the Down syndrome associated AMKLs. So you have Down syndrome associated AMKLs and you have non-Down syndromes associated AMKLs. So this one is the one which is seen in, particularly in Down syndrome. And for KMT2A, MECOM and NUC98, you can have a blast percentage of less than up to 20%, which is acceptable. So coming to the next question. So this was a 42-year-old male with history of on and off gum bleeds. IPT was suggestive of an AML with monocytic differentiation. On bone marrow, we had blasts and pro-monocytes which were present, at least 60%, and scattered eosinophils and eosinophilic precursors which were seen. Based on this, can anyone guess what abnormality it could possibly be? If I say M4EO,
Yes, M4 eosinophilic. Trisomy 8 and there might be inversion 16.
Inversion 16 trisomy 8.
Inversion 16
So this was an inversion 16. So there are interestingly, there are two types of inversion 16s. This is the one at P13, 1Q22, and there is one inversion 16 at Q24. So the one with the inversion showing the core binding factor beta and MYH11 is the one associated with good prognosis, whereas the core binding factor alpha and GLIS2 inversion 16s, they are associated with extremely poor prognosis. Okay, so yeah, this case was an inversion 16 along with an additional trisomy 8 which was seen. So what are the types of inversions that are seen normally? So this is a common chromosome with a centromere. So here there's a break. So either you can have an inversion around the centromere or an inversion along one arm of the chromosome. So that is, you have pericentric and paracentric inversion. So pericentric inversion is when there's a break of the chromosome around the centromere, and paracentric is when there's a break in the chromosome on one of the arms, that is only on one side of the centromere. So the position of the centromere remains the same. So can anyone tell me another example of any other inversion they know of, apart from inversion 16? In hemat malignancies. Okay, so we also have an inversion 3Q, which is seen. Okay, so here what is there is that G2DHE juxtaposes EVI1 and increases its transcription other than GATA2, causing leukemogenesis. So this is also associated with extremely poor prognosis. So these inversion 3Qs, these are paracentric examples. So this is seen at the position of Q21 to Q26. So here there's a break and it inverts on this side. So yeah. Okay. Can anyone describe me these pictures which we've taken? Can anyone tell me what this may be? These slender rod-like structures. Can anyone?
All rods.
Yeah, correct.
AML M3.
So this was a case where there was suspicion of an query APML. So we had seen these abnormally folded nuclei cells, the promyelocytes, and some blasts with these few ore rods which were seen. So So on IPT, this case was HLA-DR negativity. But if you always find an HLA-DR negative on APML, query APML, it's always important to rule it out and confirm it by a PML-RARA FISH study. Why is it so? So we had this case referred from Bangladesh. So this was a 27-year-old male which was treated as APML at Bangladesh, and the marrow was suggestive of APML. So the patient took ATRA for at least 3 months, post which the patient was referred to our center. So here what happened is the bone marrow of this patient was done. Although there was a reduction in the count of the promyelocytes, but the aspirate still showed those hypogranular promyelocytes. So when we did the karyotype of this patient, we found this translocation of his 1117. So this is a variant PML-RARA translocation that we see. So normally that you know of is a PML-RARA, right, which is a translocation 1517. So we also have translocation, we have variant translocation. So RARA is known to have about 45 other partners that it can have. So our FISH for PML-RARA was negative in this case, whereas we saw 3 copies of this RARA gene. So RARA is denoted by green, and we saw 3 copies of this. So that means 1 copy was rearranged, but we didn't know the partner in. So we had a RARA break-apart probe also, which showed that there's a rearrangement of this RARA gene, and which was confirmed on karyotype as we found this translocation 1117, which codes for ZPTB16. So it's just not about the variant translocations. Sometimes what happens is you need to confirm, sometimes these cases can be negative on FISH too. So this was an image from the ASH image. This was a 67-year-old male who presented with pancytopenia and had these abnormal promyelocytes which was seen in the marrow. But on karyotype, this patient had a normal karyotype, and on FISH also the APML for 1517 was negative. So sometimes we have these cryptic cases of APML which have insertions of this PML gene to the RARA gene. So these can be missed on FISH sometimes. The resolution of the FISH will not be able to catch it. So in cases of these small insertions, so it's also important to confirm them by RT-PCR, especially if you have a strong clinical suspicion and also if you see these promyelocytes on the aspirate. So here at our center, what we do is whenever we get a sample for APML, we do get, if the marrow is done, we do get it for karyotyping. If not, we at least do FISH for PML-RARA, which is done in about 12 to 24 hours. And we have a dual color, dual fusion probe for it. So depending upon the pattern that we get, we call it positive or negative. So if it's positive, it confirms APML right away. But if it's negative and we have an extra RARA signal, so we always suspect a variant RARA translocation. So in terms of this, we'll put a RARA break apart, and we'll also karyotype these samples just to know the partner of this. Or else, if not, if they are negative, also you should be confirming them with RT-PCR, where these the cryptic translocations, sometimes they can come positive in cases of RT-PCR. So everything always is not APML. For example, now in this case, it was a 40-year-old female who presented with extreme weakness, with bone pains, low-grade fever, and occasional bleeding from the gums. So the aspirate had, the peripheral smear had some atypical cells, and the marrow showed necrosis and these clusters of these round cells. So in this case, we had suspected a query of MET. We were not sure what this is, but we had also interestingly got this sample for karyotype. So when we had karyotyped this, we had got a translocation 816. So these particularly, they mimic DIC-like features and they mimic APML, these translocations, and so they have to be confirmed, and as the patient needs transplant in these cases. So when we went back to the marrow, we also saw marked areas of these erythrophagocytosis. So these translocation 816s are basically very rare, but they have a similar DIC-like profile. They will show a few APML-like cells, and they may be clinically misinterpreted as APML and treated for APML, but they are more poor prognosis, and they require immediate treatment, and they require immediate transplants, but they can relapse after transplant. So next was another, so when you have acute lukes with monocytic differentiation, what all translocations can you suspect, or what all abnormalities can you suspect? Can anyone tell me?
Differentiation.
Okay, so whenever you have any acute loop with monocytic differentiation, you should suspect for KMT2A gene rearrangements, that is your MLL gene rearrangements, especially if the patient is an adult patient with high counts. Or you can even suspect for inversion 16. So yeah, so this was a patient where we had got a translocation 911, so KMT2A rearranged. So 911s are comparatively better prognosis, intermediate prognosis as compared to the other KMT2A gene rearrangements. I had shown by Arun earlier, KMT2A houses multiple partners with multiple genes. So it is advisable to use a break apart probe sometimes because we cannot particularly target a particular translocation in these cases. So what do we do for AMLs in cases of in Tata Medical Center is we get a peripheral blood or a bone marrow sample. So of course the morphology is tested for, IPT is done for those samples. So the heparin samples which are there which come to our lab for karyotype and fish, and the EDTA samples go to the molecular lab and the flow lab. So whenever there's an AML workup, we will see the morphology. If there is suspicion of an APML, we'll straight away do a fish. Also, if fish is negative and still there is a strong clinical suspicion, an RT-PCR should be done to confirm it. We should do a karyotype and try to look for the known genetic translocations which are seen. And if still normal, then we do NGS-based myeloid gene panel analysis. Next was a 55-year-old female who presented with splenomegaly, and her HP was 8.3. Based on these findings, can anyone tell me what they can suspect?
Chronic myeloid leukemia.
CML.
CML.
Yeah. So this patient was a translocation 922. So that is your CML, that is your Philadelphia chromosome, which can be seen by this tiny 22 and a long 9 chromosome where we'll see. So interestingly, this patient was a CML patient, but it also has another abnormality. If anyone is able to pick up. So this patient also had an inversion. So this patient was actually on imatinib and he was not on any complete hematological response even after 6 months. So this patient also showed an inversion 16. So translocation 922, that is CML, along with an AML, what would you call it? Basically a CML and blast crisis.
Blast crisis.
Okay, that is blast crisis, the blast showing inversion 16. Okay. So similarly, we had this 38-year-old male who presented with fever and weakness. And so this patient was a diagnosed case of 922 CML. And due to the pandemic, the patient was not able to come to the hospital for follow-up. So after around 18 months of being on imatinib, this patient after 18 months presented to the hospital with headache, fever, weakness, and the peripheral blood of this patient showed a shift to left in the myeloid series and presence of 70% promyelocytes. So this patient was a known case of CML and also showed a presence of a translocation 1517, that is your PML RARA. So can anyone guess the diagnosis?
CML with blast.
Yeah, but in blast crisis of which abnormality? Translocation 1517?
Promyelocytic.
Yeah.
Promyelocytic.
Yeah, correct. Correct. So we also get a lot of CML patients on follow-up. So sometimes when they are not responsive to treatments, there are a lot of abnormal. Sometimes you have to look up for abnormalities if any are present. So for example, if we have this presence of an isochromosome 17, so as we all know, the short arm, that is your P arm of 17, houses the TP53 gene. Okay. So what is an isochromosome? So this is your normal chromosome with a P arm and a Q arm. So if there is absence of the P arm and your Q arm, that is your long arm, duplicates. So that is you'll have two Q arms. So this is an example of an isochromosome. Similarly, if this P arm, the long arm is deleted and there is duplication of this PM. So that is there are 2 PM. So this is an example of another isochromosome with only 2 PMs. So what happens in this is that this chromosome will not have the long arm, right? So the genes of the long arm are deleted in this chromosome. So you'll have only 2 PMs. So in cases of this isochromosome 17, this short arm is deleted and there's duplication of this long arm. So you'll have 2 of these long arms and this P arm is deleted. So that means the P arm which houses the TP53 gene has gone. So why is it important? So in a lot of these CML cases who are not responding to therapy, we do get karyotype of these patients and we have to look for particular abnormalities. So if these abnormalities are present, it just indicates that the chances of this patient going into blast crisis will be faster. So a lot of these, so this was a paper which showed the risk stratification of these abnormalities. So you will have like abnormalities like trisomy 8, loss of Y, extra Philadelphia chromosomes, and the groups which have like isochromosome 17, which is your loss of TP53 gene, monosomy 7, deletion 7q, the 3q, the MECOM gene rearrangements, as I said earlier. So these patients having these abnormalities will fare poorly, whereas with these abnormalities will will generally do a little better. So what is an additional Philadelphia chromosome? Is that so? Which is your Philadelphia chromosome? Basically, which one?
922.
20.
Yeah, 922. But which one represents the Philadelphia chromosome?
22.
Chromosome 22.
Yeah. So an additional Philadelphia is just that there is one more Philadelphia which is present. So these you'll have a lot of aberrant fish patterns which are seen. So when we use this BCR-ABL probe for detecting these 922 patterns, so we get n number of numerous patterns. So when you have this 3 fusion, 1 red, 1 green, that is this duplication of this Philadelphia chromosome. So that is an additional Philadelphia chromosome which is seen. Okay. So coming to ALLs. So can anyone tell me in ALLs which are the good risk cytogenetics?
Hyperploidy.
Hyperdiploidy.
Yeah.
Good.
Correct. And ETV6 RUNX1 fusions. Okay. So based on the how the patient presents, so the genetic abnormalities are divided as high risk, intermediate risk, and good risk. So in good risk, in terms of children, we have your high hyperdiploidy and the ETV6 RUNX1 fusion. And the high risk ones are the IM21, that is intrachromosomal amplification of chromosome 21, low hypodiploidy, near haploidy, translocation 1719, the 922, which is the pH positive ALLs, and the MLL translocations, that is the KMT2A translocation. All the other abnormalities are grouped as intermediate risk. So at our lab, what is our cytogenetic strategy that we use is whenever we have an ALL sample, so we get it for karyotyping as well as FISH at the diagnostic time point. So there are minimal number of 4 probes which we use. The ETV6 RUNX1 fusion probes, the MLL break apart probe, the BCR-ABL1 fusion probes, and we have another TCF3 fusion break apart probe that we use for classifying all the abnormalities. And also we have the karyotype to confirmation of this high hyperdiploidy, hypodiploid groups, that is for number of chromosomes, because this matters in case of risk stratification. And we also use an additional of centromeric enumeration probes for 4, 10, 17 to sometimes confirm hyperdiploid cases which may be missed on karyotype but are seen on flow ploidy if we get it as hyper, just to confirm. So our FISH panel basically consists of 4 probes, the ETV6 RUNX1 fusion probes, the BCR-ABL fusion probes, the KMT2A gene rearrangements, and the TCF3 gene rearrangements. And karyotype level, so these are the ploidies that we get in our flow plots. So we get the flow ploidy, which tells us what could be the number of chromosomes which could be seen on your karyotype. So yeah, of course, karyotype is the gold standard to confirm whether it's high hyper or low hyper or low hypo or near triploid or near haploid. So just an example. So this was a 3-year-old male who was confirmed as pre-BALL on IPT. So can anyone guess what is this abnormality?
Hyperploidy.
High hyperdiploidy, correct. So what will be, how will you risk stratify this patient?
Trisomy 4. I
Yeah.
mean, 21 million.
No, so there is a gain pattern which is seen. Okay, so this was a 3-year-old male. So your normal count would have been 46XY, correct? So,
Yes,
so you'll have normally two sets of chromosomes, right? But here there is an extra copy. Like you have gains of 4, you have gain of 6, you have gain of 8, you have gain of 14, 17,
17.
18, two 21s you have extra, and a gain of X chromosome. Okay, so this was basically an example of a high hyperdiploidy. So how will you risk stratify this patient?
Good prognosis.
Good risk.
Correct. So similarly, this was a 6-year-old female. She was also diagnosed with pre-BALL, and this was her karyotype. So can you tell me what this is?
Duplication.
Yeah. So there is duplication on the long arm of chromosome 1, correct?
Yes. And duplication.
Also, there are these gains of these chromosomes. Correct. So normally, very commonly, we find high hyperdiploid. So we'll find these duplication segments in high hyperdiploides. Very commonly, we find it. So what is duplication basically? So this is your normal chromosome. Okay, this is your P arm, this is your Q arm. Now, supposing this segment doubles. Okay, so that is, it's the genetic material here has duplicated. So this is what you'll call it as a chromosomal duplication. So this is what we see here. Can you see this dark band here? Yeah, so this dark band is also seen here as well as here, right?
Okay.
Yeah, so this part has doubled. So basically there is duplication of this segment, which we can see, which is commonly we come across this in cases of high hyperdiploides. So similarly, we had this 58-year-old females with pre-BALL with an aberrant myeloid expression on IPT. Can anyone guess what this is?
922 translocation.
So what will you call an ALL with 922?
The poor prognosis.
Poor
Yeah,
prognosis.
so you call it basically pH positive ALLs. Okay. So whenever you have BCP ALLs and it's a young child with an aberrant myeloid expression, you should generally suspect the ETV6 ranexon fusions. And in adults, sometimes we suspect an 922. So depending on, we take into a lot of factors into account, the age of the patient, the TLC of the patient, what we find on IBT. Based on that, sometimes a lot of probes you can kind of get a hint of what it can come out to be positive. So as I said earlier, we also do a TCF3 break apart probe. So why is this important is basically TCF3, commonly it has two partners, the PBX1 and the HLF. So PBX1 is seen on chromosome 1 and HLF is seen on chromosome 17. So both these two genetic abnormalities, they have different prognosis. So prognostically, if you get a 119 translocation, it goes into intermediate risk. And if you get a 17-19 translocation, it goes into high risk. And these patients do really poorly. So translocation 17-19 patients usually present with hypercalcemia and DIC-like features. Also, we do the KMT2A gene rearrangement, that is the MLL break apart probe which is used. So these so there are multiple partners for these gene rearrangements, and these are commonly seen in 60 to 70% of the infant ALLs and 15% in adults. So as discussed earlier by Arun, so break apart probe, your fusion pattern is normal. So when it splits as one fusion, one red, one green, so that means the gene has split and it has gone to another partner chromosome. So why is this important? So we had this case of a 4-year-old male who was a diagnosed case of ALL and was referred to us for further management. So cytogenetics, which was done outside, was negative for KMT2A, that is your MLL gene rearrangement. But a query MLL gene was suspected, and 2% of the cells were positive for MLL gene rearrangement. But interface cells, 2% is not a proper cutoff. So we have cutoffs for different probes. So in fusion probes, you have a cutoff of 1%. If you see the abnormality even in 1%, it will be caused as positive. Similarly for break apart, so for different probes, we have different cutoffs. So for our MLL break apart probe, we have a cutoff of 9%. So since it was seen only in 2% cells, we were not sure whether it's present or no. So they had done a marrow for this patient, and only one metaphase showed this translocation 411. So part of this 4 has gone to 11, and similarly, a part of 11 has come here. So it was seen at the breakpoint of 11 at Q23, which houses the KMT2A gene. So since we had only one metaphase which showed this, and we had only 2% cells on the interphase, we were not sure. So we do something called as match metaphase in these cases. So what is a match metaphase? Is basically this is your arranged chromosome, which was seen here. So we'll destain the slide and we'll perform FISH on this. So we will know which chromosome is 11 and which chromosome we had kept at 4. So based on this, we'll confirm what were the partner chromosomes. So in this case, we had confirmed it, and it was seen on derivative 4. That was that's how we confirmed the 4 level. Coming to the next question. So this was a 45-year-old female, and which basically has on her marrow she showed increased number of MEGs and showed features of dysmegakaryopoiesis in the form of hypolobation and monolobation. Just based on this history, can anyone guess what it could be?
PM
Syndrome deletion 5q.
But which abnormality could query be suspected?
The thrombocytosis for the most probably deletion 5q.
Deletion 5. Correct. So this was the karyotype of this patient. So we saw a deletion 5q. So can you see if I have written, here it's written as deletion 5 at Q13, Q33. So this means that it's an interstitial deletion. So what does an interstitial deletion mean? That there is a break in the chromosome, this genetic material is deleted, and these two arms are then joined back. So this is the chromosome that you get. So basically, in cases of IQ deletions, which is generally seen in these MDS cases, so we have two regions. We have these commonly deleted regions, which are from Q14 to Q33. So we have two segments here which are seen, which are the proximal segments at 5Q31 and a distal segment which is at 5Q33. So the distal segments are the most commonly ones which are deleted in the isolated deletion 5Q syndromes, and the 5Q31s are seen in the aggressive forms of MDS and AMS. So why is it important to know this? Is because these areas house these RPS14 and the EGR1 genes. So here, when this segment is deleted, there is haploinsufficiency of this RPS14 gene. There is a blockage of the erythroid differentiation, and the P53 activation which is seen in these cases. So as we all know, so this is the classification of how the abnormalities are classified in cases of MDS. So based on the prognostic subgroups, we have very good, good, intermediate, poor, and very poor. So PIQ deletions come in terms of the good prognostic subgroups. Can anyone tell me what can be used to treat deletion 5Q?
Lenalidomide.
What can be used? Someone just said it.
Lenalidomide.
Yeah, correct. So so coming to the cytogenetic aspect in plasma cell neoplasms. So we have this classification system, basically the M-SMART version, where we classify the cytogenetic abnormalities as as standard risk and high risk abnormalities. And we also have two new terms which have been introduced, the doublet myelomas and the triplet myeloma. So basically the primary cytogenetic abnormalities are the trisomies and these translocations with the IGH gene, of which translocation 11, 14 and 614 are standard risk and rest all are high risk. And the secondary cytogenetic abnormalities, which are the 1Q amplifications, the 1P deletions and the MYC translocations and deletions of 17P. So this is your M-SMART classification which is used. So any myeloma sample that we get here, so first thing that we do is enrichment of the plasma cells, and post which we have a myeloma panel for IGH gene, for deletion 17P, for 1Q amplification, 1P deletion and the trisomies that we put, based on which we'll give the abnormalities and we'll risk stratify the cases. So this was a case of a 53-year-old male, where you can see there is gain of these 5, 9, 15 chromosomes. So this was basically a trisomy which was positive, but it also showed an amplification for 1Q. That is, you can see these 4 gain copies are there. So this was basically And along with it, there was a translocation 414 which was seen. So this was a case of double-hit myeloma. We also have something called as therapy-related leukemias. So that is basically that the patient was treated for some other malignancy. For example, any solid tumor malignancy like breast, ovarian carcinoma. And after a certain time point, long-term period, the patient has developed as MDS or into a myeloid leukemia. So, for example, now this was a 30-year-old male who was diagnosed and treated for Ewing sarcoma. And he came up after around 56 months, the patient came with MDS EB2. So when we had done a karyotype for this patient, again, this patient has an MLL gene rearrangement and a translocation 1117 which was seen. So this was basically a case of therapy-related abnormality, which was showing a KMT2A gene rearrangement. Can anyone identify this test?
Break analysis.
Chromosome
Break
breaks.
Yeah. So in which case do we do it?
anemia.
Fanconi anemia.
Yeah. So basically this was a radial formation which was seen, and this is showing a chromatid break which is seen. So what is the principle of this test is basically there is impaired ability to repair these DNA cross-link breaks which are seen. Okay. So we do in cases of stress cytogenetics also we have this protocol where we'll count these number of radial formations which are seen, based on which we'll give it as positive or negative. Thank you. I'd like to thank Mayur sir for giving me this opportunity, and Sumit sir and conducting this workshop. Any questions?
So this was Aishwarya actually want to introduce herself. So this is Aishwarya Dabe. She's a senior final year fellow in cytogenetics. She is a pathologist. Any questions?
Sir, I would like to ask a question. Dr. Aishwarya, you said that something called as interstitial deletion that was there, right?
Yeah.
So what does that mean exactly?
So we have two types of deletions. We have a terminal deletion and an interstitial deletion. So for an example, if this is your short arm and this is your long arm, okay? So interstitial deletion means this part is deleted and the remaining part which is there is joined here.
Okay.
Okay. Yeah.
Something from the center which is gone.
Yeah, so this part is deleted and the remaining two segments have joined back to give this chromosome. So this is an interstitial deletion. If it's a terminal deletion, then there'll just be a, supposing if here is, if this segment is deleted and this is joined back. So this
how
is a
do
terminal
you know,
deletion.
looking at the chromosome, if it is an interstitial deletion?
Yeah, so we have this banding pattern. So so we have to check the number of bands, how it presents and how it looks to call it whether it's a terminal deletion or an interstitial deletion.
Thank you. There are a few more questions in the chat. So, Karishma Khatri has asked, can you please tell more about tricolor probes?
So we have so tricolor probes in cases of plasma cell neoplasms that we use commonly is for 5, 9, 15. So basically in every chromosome, so you'll have two sets of chromosomes, right? One from the mother and one from the father, right? So if there's an extra pair, extra gain of chromosomes, so we'll have call it as an high hyper. So
basically,
can you
sorry, I'm just pitching in. So you know the tricolor probe means, so usually you see when we do fish, we have an aqua red and an aqua green filter. That's why you're seeing red and green, right? I mean a spectrum red and spectrum green, right? And you also have a spectrum aqua, right, which is blue. So basically, this is nothing but using a probe design which has three different colors of fluorescent probes: red, blue, and green. So these are called tricolor probes. So here you can actually look at abnormalities of three loci. Those three loci could be three chromosomes, those three loci could be three genes. And so it's basically a way of designing the probes, because sometimes you have to deal with more than two genes and you want to look at three genes together, and or maybe look at 3 chromosomes in one go, and that is where you use the tricolor probes.
Thank you, sir. Dr. So, G. Sandhya is requesting to elaborate more on inversion 16.
Inversion 16.
Like the breakpoints and everything.
Yeah. So inversion 16, as I said earlier, so basically there are two types which is seen, which is core binding factor beta and core binding factor alpha. So the ones which core binding factor beta with MYH11 as the partner, so these are basically pericentric inversions, right? So the breakpoints are different. So if it's P13 and Q22, so that is core binding factor beta with MYH11. And if it's P13 with Q24, it is core binding factor alpha with its partner as GLIS2. So the ones which involve the core binding factor alpha, basically they have a poor prognosis. And the ones which involve the core binding factor beta with MYH11 fusion, these are good prognostic features.
Thank you so much, Dr. Aishwarya. We may now proceed. Thank you so much.
There are no more questions. We go to the next speaker, which is Dr. Karth. He's also a second year fellow in cytogenetics. He's a pathologist, right? So Karthik will talk about fish in solid tumors.
Good afternoon, everyone. I'm Dr. Karthik, a fellow in Department of Cytogenetics in the Tata Medical Center. So I'm going to be presenting on the aspects of fish in solid tumors. So as pathologists, in solid tumor assessment, we would be knowing that we get a solid tumor tissue from which we prepare slide paraffin blocks or FFPEs, and then from which we get sections cut on the slides, and then we apply stains or IHCs to reach a diagnosis in this workflow. But sometimes what happens is that even in spite of the IHC, we may have a diagnostic dilemma. At this point, we use fish in certain cases to help us prepare the report of a solid tumor case. So what is the difference between the IHC and fish? IHC, or the immunohistochemistry, as you know, would assess the presence of a protein or the antigen expression using antigen-specific antibodies. Take example, HER2 IHC, or we have ROS IHC. But when it comes to fish, it assesses the presence of chromosomal or genetic aberrations using specific fluorescent tagged antibodies. When it comes to genetic testing in cancers, we have cytogenetic testing and molecular genetic testing. In cytogenetics, as you all know, that we have karyotyping and fish, which can be generally done for cancers. But when it comes to solid tumors, especially for first in karyotyping, as Mayur sir had told in the initial part of the presentation, that karyotyping requires live dividing cells to be cultured to obtain the metaphases, a longer turnaround time period. Thereby, karyotyping is very difficult to perform in solid tumors, and it's not done usually. But when it comes to fish, it does not require live dividing cells strictly. Interface cell analysis is enough for interpretation. And one main advantage is that it can be done on the solid tumor tissues, that is FFPE tissue, even on cytological smears and imprint smears. And has a very shorter turnaround time period. Thereby, this becomes a routinely preferred cytogenetic method of testing in solid tumors. The other important reason why we need fish in solid tumors is that it has three reasons. One, it can be used for diagnostic purpose, one for therapeutic purpose, and one for prognostic purpose. Firstly, in the diagnostic purpose, we know that fish can be used in the diagnosis of various soft tissue sarcomas, oligodendrogliomas, where 1p19q code deletion is important, lymphomas, especially certain types like mantle cell lymphoma, follicular lymphoma, and anaplastic large cell lymphoma. And when it comes to therapeutic purpose, in breast carcinomas, fish for HER2 new status is a very important marker. Then in lung adenocarcinoma, ALK and ROS1 status is an important marker. And in the prognostic purpose, it is very important to differentiate between double hit and triple hit lymphomas. Then in medulloblastomas, where NMIC amplification is seen in group 3 and group 4, and then in neuroblastomas. Yeah. So before we just see a case, I'll just give the brief intro of the case. So we have a 52-year-old female who has a left breast mass, whose histopathological examination is suggestive of a invasive carcinoma, invasive breast carcinoma, ER positive, PR positive, and the HER2 was equivocal. So what test would you, or what fish test would be performing in this case?
Fish.
Fish
Yeah, fish.
amplification test.
Yeah, correct. So we would be performing for fish for HER2
Fish
amplification.
amplification.
So what is HER2? So HER2 is a transmembrane receptor which has a receptor tyrosine kinase activity. As you can see here, it is a gene which spans the transmembrane across the membrane and has a tyrosine kinase activity. Thereby, it's a receptor tyrosine kinase, and it generally dimerizes with other receptor molecules. And upon this dimerization, there is an activation of the tyrosine kinase activity of the molecule, which would phosphorylate downstream pathways like SOS, RAS pathway, MEK pathway, MAPK pathway. So further downstream activation of these pathways leads to cell differentiation, migration, cellular proliferation, and other effects. So when do we do HER2 fish in solid tumors? So in the case of invasive breast cancer, which has an IHC 2 plus or an equivocal IHC staining for HER2, those are the cases when we do the HER2 fish according to the ASCO guidelines 2018. And how do we score in our lab? So we use the Cytovision Cytolite dual color probe. So in which the green color as shown here targets the HER2 gene, and the red color signal targets the centromere of the chromosome 17. So this would be scored by two readers, where two readers would be scoring 20 cells each, and both the readers would be giving the average of green and that is HER2 copies and average of the centromeric copies, and ultimately derive the ratio of the HER2 over the centromeric 70. And according to the ASCO guidelines, we will interpret it as positive or negative for amplification. Certain important points to be noted when we do any solid tumor testing is that since we do it on FFPE blocks, we obviously would be having corresponding H&E stain slides. So we need to have a relook at the H&E slide to correlate with the area of tumor cells while counting. And when we count for HER2 in invasive breast carcinoma, only the invasive component is to be counted. And the DCIS component should not be counted when we assess for the HER2 in breast cancers. So what are the criteria we call it positive or negative for amplification is that if the average number of HER2 copies is more than 6 copies per cell, it is HER2 amplified. Or if it is average HER2 is less than 4 copies per cell, it is negative. But if there's a borderline situation where the average number of HER2 copies is between 4 to 6, we look at the ratio mainly, that is the HER2 by the 17 ratio. If the ratio is more than 2, then it is considered as an amplified tumor, HER2 amplified tumor. However, if the ratio is less than 2, then it is non-amplified. So here's an example of an image which shows a case of amplified tumor. So as you can see here, these are the individual tumor cells. And as the key shows here, the labels, the green is the HER2 gene, or the HER2 signals, and the red is the centromeric probe signals. So you can see that there are multiple copies of HER2 or the green signals, and This is a positive tumor, HER2 amplified tumor. And another image which shows just almost 2 copies of HER2 and 2 copies of centromeric probe 17. So as I told you, the average number of copies of the green would be less than 4 in this case. So it is a non-amplified tumor. But then in some cases, we encounter a phenomenon which is known as intratumoral heterogeneity. So what do we mean by that? So intratumoral heterogeneity is defined as a phenomenon where there are presence of 2 or more distinct clone of cells within a single tumor mass and can be characterized by any of the following features. So firstly, there can be discrete populations of both amplified and non-amplified tumor cells, or there is an intermingling of the amplified and non-amplified cells together, or there is a certain small amount of amplified cells in a predominantly background of non-amplified tumor cells. So I would be showing you an image next way. So how do we report this intratumoral heterogeneity? So as I told you, when we see the H&E slide and correlate with the fish, so if we see that the amplified cell component is more than 10% of the tumor, but it is less than 50% of the overall tumor, then we count these amplified tumor cells and mention them in the report as amplified tumors. So basically, whenever there's an intratumoral heterogeneity, it means that there is a amplified tumor component which is present in the tumor mass. So this is an example here. So this was a case where we had initially seen that there were a lot of, or predominantly the tumor showed non-amplified cells, where there were the average green copies were between 2 or 3. That was, it was less than 4 predominantly. But then when we scanned the whole slide, we saw that there were these large tumor cells in the same case where it had amplified copies of HER2 in the same case. So thereby, this was a case which had intratumoral heterogeneity. What is the significance of this HER2 status? So as I told you that HER2 was a transmembrane receptor which heterodimerizes with the other receptors and thereby activating downstream pathways. So when this HER2 gene amplification is present, this HER2 gene amplification results in an overexpression of these HER2 receptors on the cell membrane. Because of this overexpression of these HER2 receptors, we have excessive dimerization of these receptors, which leads to an increased activation of the downstream pathway. Thereby, we have the oncogenesis of the breast cancer. So what is the therapeutic aspect which is very important in this case? So targeting this HER2 amplified tumors, we have a molecule which is Herceptin, or otherwise known as trastuzumab. So how does this Herceptin act? So the mechanism of Herceptin is that it leads to an blocking of this dimerization of these HER2 receptors. As I told you, the dimerization of these receptors would lead to the downstream activation of the pathways. So this trastuzumab would block the dimerization of these receptors and thereby would stop the further downstream signaling which is responsible for the cancer cell growth. So as you can see here, we have molecules which targets this HER2, like most importantly trastuzumab, that is Herceptin, or then we have other molecules as labeled here. Next up, we would be seeing what is about ALK and ROS. So ALK and ROS, what are ALK and ROS? Like HER2, ALK, ROS, as well as the other molecules shown here, MET, RET, EGFR, all are transmembrane receptor molecules which have a RTK, that is the receptor tyrosine kinase activity. And similar to the HER2 molecule or the HER2 receptor, they have a receptor tyrosine kinase activity which would activate the downstream pathways which is responsible for the cell growth and cancer cell growth and cancer cell division. So when do we test for ALK and ROS fish? When we have a case of non-small cell lung cancer which is suggestive of adenocarcinoma on histopathology examination, we have to perform ALK and ROS IHC testing. If the IHC result is positive or unequivocal staining, then the fish testing has to be performed to rule out the gene rearrangement. And And what is the guideline to report for ALK and ROS fish positivity? So as the diagram shows and the slide shows here, that we have kept a cutoff of more than at least 15% of cells showing the break apart pattern to confirm the presence of ALK or ROS rearrangement. So one case here which shows a case of a 45-year-old female who's a non-smoker, had a mass in the left upper lobe of the lung. The HPE was suggestive of moderately differentiated adenocarcinoma of primary pulmonary origin. And IHC on this case showed an equivocal stain for ALK in this case. So fish for ALK was done, and as you can see the diagram here. And if you had remembered from the talk of Arun, how the break apart signals or the break apart pattern is positive. So in the case of normal cells in a break apart probe, ALK is a break apart probe. So we see too few signals in a non-rearranged case. But here, as we see, we can see cells which show one fused signal and one green and one red signal which is separately split apart. So this is a case where ALK rearrangement was positive. In another case, we have a 58-year-old male, again a non-smoker, with moderately differentiated adenocarcinoma of primary pulmonary origin. Here in this case, the IHC was strong positive for ROS1 IHC, for which fish testing was recommended. So again, ROS1 is also a break apart probe. So as you can see here, tumor cells which show one fused signal and one green and one red signal which is separately split apart, like here, here, and also here. So this is a case where ROS1 rearrangement was positive. And this case, this was a 67-year-old male who is a known case of lung adenocarcinoma, had a fish previously for ALK which was positive. Because of this ALK positivity, the patient was treated with a particular molecule known as crizotinib. In spite of the treatment with the crizotinib, the patient had a persistent progressive disease. So then we had done a fish for a gene named as MET, fish for MET. So basically we look for MET amplification here. And as you can see here, these are tumor cells which show an amplified green signals, and green here was MET and the red was the centromere 70. So this is a case where MET amplification was positive in a known case of lung adenocarcinoma targeted therapy. Now, what is the significance of this ALK and ROS status? So the therapeutic aspect of this ALK and ROS positivity is that we have particular molecules which is FDA approved as well as under clinical development for this lung adenocarcinomas with ALK or ROS positivity. The most common molecule being crizotinib, which is used in practice nowadays. And we have other molecules like lorlatinib, ceritinib, and various other molecules which is in clinical practice. Then we will be moving on to soft tissue sarcomas. So if you be knowing that in soft tissue sarcomas, the cytogenetic abnormalities are divided into two groups. One is tumor-specific abnormalities, and one is where we see multiple abnormalities. So in tumor-specific abnormalities, we see commonly translocations are more common, and then we have cases where there are multiple and sometimes complex abnormalities. For example, we have chromosomal translocations like EWSR, FLI1, SSX1, SS18 fusion, PAX3, FOXO1 fusion. Then we have EWSR, WT1 fusion. As well as we have certain molecular like activating and inactivating mutations in KIT, PDGFRA gene and SMARCB1 gene. And then we have multiple abnormalities which can be seen in a case like MDM2, CDK4 amplifications, which can be seen in liposarcoma. So when it comes to fusions in soft tissue target sarcomas, we have a long list of fusions seen in soft tissue sarcomas, which you can refer to the literature and get to know. And when it comes to EWSR1 gene, EWSR1 gene has multiple fusion partners, which leads to multiple histotype of tumors, as shown in the diagram here. So they are basically divided into two groups. One is the classical Ewing sarcoma fusions, so which is translocation 1122, which leads to a EWSR FLI1 fusion most commonly in 85 to up to 90% of the cases, followed by EWSR ERG fusion or ERG fusion in up to 10% of cases. The other partners shown here are rare. And then we have another group, which is the Ewing-like sarcoma fusions. So where EWSR can fuse with non-ETS partners, as shown here, NF80C2 and PATZ1. As well as we have this chick rearranged sarcomas or BCOR rearranged sarcomas. So this was a case of a 14-year-old female who had a right thigh swelling for 1 year, and the HPE was suggestive of an angiomatoid fibrous histiocytoma. As you know, AFH has EWSR1 rearrangement is seen. So FISH for rearrangement of EWSR1 was advised. And as you can see in the images here, we have tumor cells which show a rearrangement of EWSR1 gene. Here. And another interesting case where we have named it as a tale of two cities. So this is a 60-year-old male who had a swelling in the right upper arm for past 3.5 years, and there was no history of any antecedent trauma. The patient was noticing that the swelling was increasing in size since 2018, and there was an MRI which was done for this lesion, and the patient was evaluated at Chennai, where a core biopsy was taken from the lesion. So at Chennai, the diagnosis which was given was a complex cystic mass suggestive of a mesenchymal tumor with a hemangiopericystoma pattern. The positive for CK, FLA1 and CD99. However, review at one of our labs at Mumbai had shown had reported as a tumor with clear cell morphology with strong expression of PAG8, favoring a diagnosis of metastatic clear cell renal cell carcinoma. Subsequently, the patient was operated at Chennai, where final diagnosis was given was sclerosing epithelioid fibrosarcoma, and for which a first gene rearrangement FISH was advised for confirmation. So this is the FISH of first gene. Can anyone comment like if it's a positive or a negative pattern?
So what do you guys see here? Just say what you see. Don't say positive or negative.
Yeah.
It's a fusion. It's a fusion gene in C.
So you are seeing fusions.
Yellow.
Yellow.
Yellow fusions you are seeing, correct? So it is a break apart probe. So seeing a fusion is positive or negative?
It's negative.
Negative. So it was negative for first rearranged.
Okay. Good. So it was negative for first gene rearrangement. And then we had also done FISH for EWSR1 gene rearrangement. And as you can see here, we are seeing single fused signals in all tumor cells. So this was also negative for EWSR1 gene rearrangement. Yes. So this was a tumor cell morphology which was seen, where we had cells with clear cytoplasm and an epithelioid morphology or epithelial to spindle cell morphology. And just for a reference, this is usually how we have a case of clear or the histomorphology of clear cell renal cell carcinoma. And these were the IHC findings in this case, where we had following IHCs. And a FISH, in this case, a FISH for SS18 rearrangement was done. And you can see here that there is a breakup, and SS18 or synovial sarcoma 18 FISH is also a rearrangement probe. So as you can see here that we are seeing tumor cells which is showing a rearrangement pattern. Yes. So this was positive for SS18 rearrangement. Another image showing the positive for rearrangement pattern. So based on the IHC and the cytogenetic features, diagnosis of poorly differentiated synovial sarcoma was given. And the case reviewed at Sanjay Gandhi was also Singapore General Hospital was suggestive of synovial sarcoma. And next we will be seeing on MDM2. This was a case of a 70-year-old male who had a soft tissue tumor over the left arm, for which MRI was done, showing a large intramuscular lesion involving the left brachial muscle. Histopathology was suggestive of atypical lipomatous tumor. A FISH for MDM2 was done in this case, which was suggestive of a MDM2 amplification. So this is the criteria which we use to report MDM2 amplification. So we see the number of signals of the MDM2 over the centromeric probe 12. And if a ratio of more than 2 is seen in more than 10 nuclei, we call it as positive for MDM2 amplification. Now, what is the significance of this MDM2 amplification? One of the most important things is that we use it to differentiate well-differentiated liposarcoma, atypical lipomatous tumors, as well as de-differentiated liposarcoma from the histological mimics. And then it is also proven to be one of the proposed mechanisms of chemotherapy, radiotherapy, and TKI resistance, as well as immune checkpoint therapy resistance. This MDM2 amplification, it's a founding genetic abnormality. So it can be seen in well-differentiated liposarcoma as well as de-differentiated liposarcoma, where in spite of the de-differentiation of tumor cells to any morphological type, the MDM2 amplification will be persistent. Next up, it's a case of a 3-year-old male who had an abdominal mass. Histopathologically, there were small round blue cells seen in the histopathology, and the IHC of the tumor cells had showed a diffuse cytoplasmic positivity. And we can see, can anyone guess what was the FISH probe used here in this case?
The MIC amplification.
Yeah.
MIC amplification.
So, FISH for NMIC amplification was done in this case, and this shows, this image shows tumor cells showing the NMIC amplification. So, a diagnosis of neuroblastoma was reached in this case. Yeah. And next up, we have a 52-year-old male who showed who had a left frontoparietal intraaxial space occupying lesion. The histopathology of the tumor showing a fried egg appearance. The IDH-R132H IHC showing a mutant pattern of staining, and the ATRX IHC showing wild type or the tumor cells retaining the ATRX staining. So in this case, what is a FISH to be done next?
1p19q deletion.
1p19 co-deletion.
So we have to perform FISH for 1p19q co-deletion. And as seen in the images here, so we look for 1p deletion as well as 19q deletion in the same tumor cells. So in this diagram, in this image which is seen here, 1q is tagged with green color and 1p is tagged with red color. So as we can see here that each tumor cell is showing 1 red, 2 green, 1 red, 2 green, indicating that there was deletion of 1p in the tumor cells. As well, we added for 19p and 19q assessment was done. So 19p is tagged by the green color and 19q was tagged by the red color. So we can see here that these tumor cells again shows 1 red, 2 green, 1 red, 2 green, indicating that 19q was also deleted along with 1p in the tumor cells. So there was positivity for 1p and 19q co-deletion. So what is the final diagnosis here? So final diagnosis of oligodendroglioma, IDH mutant was reached here. So this is all the cases which we had seen in the presentation, and this is just to highlight the list of solid tumor FISH probes which we use at our center here. So we have one for HER2 new amplification. For lung adenocarcinoma, we do ROS1 rearrangement, ALK rearrangement, MET amplification, NDRK rearrangement, RET rearrangement. For lymphomas, we have CMYC, BCL2, BCL6 rearrangement. We have probes for translocation 1114 to confirm mantle cell. translocation 14-18 fusion to confirm follicular lymphoma. In CNS tumors, we have 1p19q co-deletion probe for oligodendroglioma, CDKN2A 2B deletion for CNS grade 4 lesions, then c-MYC and n-MYC amplification for medulloblastomas. For soft tissue tumors, we have the following probes: EWSR1, first rearrangement probe, SYT rearrangement probe for synovial sarcoma, FOXO1 probe for rhabdomyosarcoma, MDM2 amplification probe for liposarcoma. As well as we have for other tumors, we have TFE3 rearrangement probe for the MIT translocation associated renal cell carcinoma and an n-MYC amplification probe for neuroblastoma.
Okay, this we've already covered. I thought this will be a post-workshop. And thank you, everybody. I would like to thank all our colleagues from Cytogenetics. So 4 or 5 of us were just the representative. So this is our team. That's Pranay, that's Samika, Arun, Shipra, Tuhin, and of course, Aishwarya Karthik. And yeah, of course, that is me there. And Sandeep is our housekeeping staff who keeps us sane, actually. He takes very good care of ourselves, gives us very good coffee. And would like to thank Dr. Gujral, Bojes, and Bojes Madam again for giving us this opportunity. And if there are any other questions, we are most, so Aishwarya, Karthik, and me, all 3 of us are here. We are happy to answer any questions.
Okay, done. So I have a question, sir, here. So what is the problems you face in day-to-day reporting? Like for example, in flow cytometry, we have this dead cells or a debris which actually non-specifically bind with some antibody. So are there any such things in routine reporting in the cytogenetics?
When it comes to, so as you told, so when it comes to solid tumor or even liquid tumor testing, when we get sample, in case of solid tumor fish reporting, initially it was told that